What up? What up? All right. We'll wait for some people to come in here. Sorry about a minute or two late, guys. But I had to eat. I had to eat. So, oh shit, on Twitch. Let's go, Carter. That's what's up. It's cool. It's cool. Oh yeah. See, we can see on the screen like where they're, what app they're on.
Yeah.
So he's on Twitch, YouTube, Facebook. Not too many people usually.
What's up with that?
Let's wait for some more people to get in here. Give them a second. Make sure we're good on this page. Should be. Hold on. Let's see. I see you got all platforms. I see you. Are we on Instagram? Do I need to recheck that? Hold on. All right, so we're good. We are on Instagram. All right, perfect. So I see you guys. Don't say ain't swing through. You the fool. Stop playing, NC. Stop playing, dog. Oh, I can hear myself. So I see you guys. Yeah, come over to YouTube, though, man. So everybody, listen, I'm going to type it in. I can't pin it on the YouTube, though. Let me see if I can put it across the bottom of the screen. And then we're going to get started. So make sure you guys come over here. Here it is. So that banner at the bottom of the screen now. Yeah, I see you, fool. I see you, NC. Just saying. So, yeah, what up? Shout out to Tony. He's going to come on with me on Saturday. We're going to do some giveaways. Just get that out of the way first. I see him over there. So, Tony, the YouTube, this is how we do it now. YouTube, Twitch, Facebook, and obviously Instagram. And we'll do the giveaways like this as well. So make sure you follow the YouTube, guys. Danny Dreams, NC, Canna Frizz, the green one, All Stars, Magpie, East Coast Farming. All you guys come over here because if you have questions, I say it every week. I won't be able to see them over here. This way we can throw them up on the screen like that. Ade can answer your guys' questions. We're going to get into tissue culture. I'm going to have her for a little while. I think Kind of Research is going to join in at one point or another. But, really, we have at least an hour. We might have more. If she's ready to go in an hour and we're through the questions, that's fine. And Kind of Research, we'll see if he, when he gets here. But in between that time, I'll do some giveaways. So just know that, we'll make sure because Boro Florist and Snoopy, they won't be here until 10 o'clock. So we basically have a 2-hour window with her and Kind of Research, but I'm not going to keep her at 2 hours unless we really get into it and she, graces us with her presence for the next 2 hours, which I wouldn't mind at all. So, but we'll see how it goes. So, another one in the comment, in the Instagram, Beth, look at the YouTube scroll. Come over to the YouTube. Everybody come to the YouTube. All right. We got about 25 people in here. All right. So you want to introduce yourself? We can start there again. Let's start there.
Yeah. So I'm Ade. I'm a geneticist, tissue culture, pathogen testing person here. I consult on the side within tissue culture, as well as how to go around and test your genetics for pathogens. But for today, I'm going to be showing you guys how to extract DNA to test for pathogens. I can only do the first part because it takes like 8 hours to do the whole thing. So I'm just going to start it for you so you guys can kind of see how it's done, and I can even show you the equipment so you can uh, get a better understanding of the different methods before you actually should TC.
Let's go. Let's go. Let's go. Let's go.
All right.
Yo, I just want to say I'm always happy to have you on, man. I've really gotten to know you a little bit since the last time, especially, and yeah, it's just a pleasure to have you on because I feel like there's so many people that, they hear about tissue culture or HLBD testing, male female testing, all that. So she does all those things, and I want you guys to really, no question is a bad question. So no matter what it is, she will have an answer for you. If she doesn't have an answer for you, she will find an answer and she'll come back and give us the answer. So, feel free, guys, to ask anything you guys want. Tissue culture, we're going to get into HLVD testing, like she said now.
Yeah.
Questions about plants, how they look. I'm not going to give you guys questions to ask, but I feel like there's got to be a lot of questions you guys have because I always have questions on this. I'm not an expert. I know very little about it. We talked about the Mary stem last time. She could talk about that a little bit. I just feel like this is a really good time for you guys to get a little bit versed on tissue culture, HLVD, what to look for even in your plants, where to test, how to test, all that. So feel free to ask whatever questions you guys have. And I'll probably say this again every like half hour for the new people that come in. But yeah, I see in-house. I see you over there. I see you over there. You're supposed to come to the YouTube. See the scroll down there?
To the dark side.
To the dark side. She already on the dark side, man. We just gotta get her over to the YouTube.
Yeah.
That's right. Let's go. Okay, so listen, Frost Pops. I have no problem reading from the Instagram, but it makes my life so much easier because look, I have to look down to see the Instagram. On here, we can see the YouTube, we can see the Twitch, and we can see the Facebook. I could put the questions up on the screen like that, and we could all see the questions. If I'm looking down, it is what it is. I will try to glance down as often as possible, but just know I'd rather you guys be over here so I could just look at the screen and not try to look at two different things at one time. So anyway, Frostpop did have a question. His question is, how long can a plant live in tissue culture?
So it all depends on the strain itself and the genetics. When you first try and put something in TC, if you can, always try and get it tested so at least you know what you're working with. Once you have that information, then I would try to move carefully, but it just all depends on your genetics. If the plant you're trying to TC is like sick, you would have to move it a few times before you can actually just leave it, but you have to maintain them every 3 weeks, just because the PGRs, the plant growth regulators, cause them to produce cells sometimes a lot more than others depending on the strain.
What is the average, like, what's the average time, like, before you need to switch it up? So we could talk about, well, I'm going to let you do what you're doing. We can get to these, why don't you do what you're doing? Okay. Let's focus on that. I just happened to see the question, but we could revisit that question in a little bit, Okay.
All right, let me try and turn this. So here I have, wait, let me make sure my screen is clean. I've been running around all day, but here I have 3 different, I have one strain and another strain, but I have 3 different samples I'm going to take. From this chimera here, I will be taking root samples to test for hop latent viroid as well as leaf. And those will be the 2 strains here. And I also have some super berry freak that I am going to be testing just the leaf to see if it has traces. I kind of forgot the roots. So we're going to see if this will work and exactly how accurate the test will be compared till tomorrow. I'm going to retest the roots to see the 2 different results. Hell yeah. But these, you have to really, really, really um, clean the roots when you're doing this. Let me see if I can get a better picture without spilling it. You have to
Here, I'm
really
gonna do
clean
it.
the roots when and remove all the aggregates of soil from it. And it's a process. It took me like two hours to just remove the soil. Um, and I have that here. My husband suggested I use a pipette to kind of take the soil off because you can see some of this has still pieces of it, and I've been swooshing it for like hours.
So basically, you're going to test different parts of the plant for this. That's why you're showing that,
Yeah,
right?
yeah.
Okay.
So a lot of literature or like papers that are published from scientists doing research on hemp and cannabis, a lot of it shows that when a plant's more mature, the hop latent viroid, because it's latent. The whole part of latency is it's sleeping and dormant, and it usually wakens up as it matures. So taking samples of the root is suggested over leaves, just because hop latents and usually in soil, so that it gives more of an accuracy with the plant. A leaf still can give you, an accurate test, but it's just suggested to do the roots.
So when you say that, like you're saying that it's in the soil, like you said, it's in the soil. So
Yeah.
we can find it in the roots, right? So in the roots. So you want to get rid of the soil, obviously, but you're testing the roots and where else? Like, if someone was sending you in a test sample, for example, where would you have them take their samples from? Would it just be the roots, or would it be also?
If their plant hasn't reached maturity all the way, I would probably ask for a leaf and a root just in case, um, just to be sure. But a lot of the papers that I've read state that if the plant is mature, just to take the roots, and it gives you a more accurate testing. But
Okay,
sometimes people want to test before it, wakes up during flower. So you
okay.
can test the leaves. It's still accurate. It's just more suggested for the roots.
So I was curious because I've heard, I have heard when people are doing HLVD tests and they're saying take from the roots, the leaf, and maybe even like two different parts, like a different part of the plant, like maybe like this side of the plant. But that's not the case necessarily. Okay.
No, no. So if the plant has it, it'll be in the whole plant. It will express in the whole plant. Like a viroid just needs a living host because it's a virus. A virus has to have a host to attach to. And with the plant, once it has it, it has it.
Okay.
Some
So
some sometimes it doesn't express it, but I think if you go and clone the said plant a few times and it starts to get stressed out and the vigor goes down, then it might start to express it.
okay, so here's like a weird here's like the okay. So you're so it's set okay. What I'm thinking is this, right? Why go through the trouble to test the roots and clean them when you could just take a leaf sample? If the whole plant's gonna, I mean, like just total ignorance on my end. I'm just,
No, it's never ignorance. No question is
well,
ignorant.
I'm just,
Yeah. But a lot of the research that has been done when it comes to testing for pathogens, a lot of the data shows more accuracy in the roots versus the leaves, especially when, like I said, when it reaches maturity. So I would say like teen and up. That's when you would test to see if it had it. But a lot of the research that has been done, the data shows that it's more expressed and it's easier to test in the roots versus the leaves, because sometimes in the leaves it, it doesn't. Um, I don't know if it's not doesn't show, but when you run the pathogen test, it's looking for certain signals from the DNA to say
okay,
that that plant has that. And I think at a more younger stage, it doesn't express as much.
okay,
It's yeah.
let's go. So, okay, I see the roots holder data.
Yeah.
Yeah, this is why the conversation. I feel like I just I mean, I know what I know, and a lot of it, like I have a really good memory. So the conversations we've had, I just remember, I soak it up. Um, but I always, this is why I love to do this, because I'm always learning too, and I don't mean ignorant. I just mean in the fact where I just don't know, like I just don't know. So I like to ask questions. Usually I'll let, these guys ask questions. I don't see so many questions, but I'm always going to throw in my little 2 cents and see what's up too. So, all right. So we're at the roots now. So we can,
So if we could take samples of each one so I can show you. Babe, come hold my phone. Hold it like right here, okay? Mm-hmm. So let me make sure to clean my hands one more time. Mm-hmm. So with this chimera here, I'm going to, I have a leaf inside of a tube with water, and it's just a leaf from the more mature part of the plant. And we're going to take this here. It stamps a hole in it, in the leaf itself. So you just kind of hold it open like that and push down. And then you're going to take this piece here. It's supposed to come right off of there. All right. It's there. And then you kind of place it in here like so. And then you'll want to take something really sharp. I usually use toothpicks, but this is just for example. And you just shove it in there. And then you kind of close it for now. And then I'll move on to the roots. The roots is a little bit more difficult.
So then we are also, we are also testing the leaf as well then, right?
Yeah, so for the chimera, this morning was kind of a little crazy. I was running around like a chicken. So I forgot the roots for the super berry freak, but I got the chimera in the car. So
have you tested any of the chimera yet?
Like, no, this is the first time.
Let's go.
So
I'm interested to see this one. I'm very interested to see that. I am.
Yeah, I am too. I have it growing. It's in flower right now at home. It's looking really beautiful. So just make sure that everything touches the heat before touching your sample.
And how long does the test take you, did you say?
So the first part of the test takes 4 hours. The second part's like 4 to 5.
Gotcha. Okay. So we won't be able to see the results from this tonight, but we can get you to give us an update that I could post or something.
Yeah, I'll post it and I'll tag you on it. But so this is the main root here. So I kind of want to take from that because I feel it's the most mature point. Move it this way. Here, this is the main piece of the root with the most maturity. This was a side root. And to me, I don't feel there's a difference, but why not try? So I'm going to take 3 of these pieces and put them in a tube.
And how long, this is the new machine, right? This is
this is the baby.
This is the baby. So this is the baby that we were talking about last time. Yeah. That you were about to get.
Yeah, and I got her, and I get nervous to use her because she's so expensive.
I mean, isn't that what all expensive toys are like?
Yeah.
Right? For real.
You want to play with them, but you're too scared. And so it's in here, the root. That's as big as a piece that I got out for that one. And now we're gonna pull the Super Berry Freak. I'm getting extra tubes right now. I can't really touch too much stuff because contaminants.
Right.
Um,
So do you switch like well, I guess yeah, you would switch. Do you switch gloves? Do you sterilize knives? Do you switch knives? You want to tell them a little bit about like that, you know, sterilization process too?
so I use ISO. I just kind of usually put it on something to pump it on my hand, but I don't have it. I
Okay.
just put a little bit, you know, just get the tips. I washed everything down with bleach prior because of the smells. The two gases in the air are really dangerous. Um, but I always make sure everything's clean. We're sprayed with ISO from top to bottom at least 3 times, 91%. And
Sure.
then everything that we touch that we come in here with is completely contaminant free. If it's not, you can't be in that area.
Okay.
Because if any little contaminant goes into this DNA extraction, it can give a false positive, false negative, or a blank reading.
And how much is the testing? Like, if someone, well, I'll let you finish. We can talk about that soon.
Okay, here. I'll um so this alone is $700.
Dang. So what is that exactly?
So this is nucleo. I can never say the word, but it basically it's free of like nuclease. It's like DNA.
Nuclei,
Free. Yeah.
nucleize,
Yeah. I
something
can
right?
never say that word. I always want to say it in Spanish, but it's
Yeah.
free of any like DNA. And
Okay.
then denatured alcohol. These two are needed for DNA extraction. And then this here is a master mix for 200. This is like $1,200.
Okay.
So all my chemicals together for the machine were $5,000.
Okay. How long will they last you? How long will those chemicals last you?
Um, not too long at all. Um,
Really?
yes, they have expiration dates. Um,
Okay. Well, like, wait, hold on. Let me turn this on. So, how
so
many tests will you be able to do with that? Like,
so this one is for 200. So this whole thing right here just removes the pathogens that are on here. There's certain buffers. A buffer is a chemical that will signal the machine to kind of enhance that area of the DNA to kind of pull it forward so that the machine can actually take a picture of it. But this, let me see if I can show you. This is pretty much what it is. There's very little bit in there. It's like the tip of it.
just the tip. So, how much was it? How much were they again? Just that?
So this alone was 12, and then some of these were like $2,000. It just depends on what chemicals, but altogether for hold on 1 second. Let me get in mine.
And I see you have a thermostat there too. So, what is
Oh
it?
yeah.
Is that the temperature inside there right now?
So this is set as Celsius because it has to be -2 Celsius, and right now I shouldn't have it open, but
Okay. Okay.
usually it's -1 Celsius so that all my chemicals stay cold, and because DNA kind of sits still at a certain temperature. But these were some that I extracted. This is some DNA. Wait, this is a DNA. It's
So, how long once you extract it like that? That's already done extracted. Like, can that be tested now, or does that?
yeah. I'm
And
trying to
how
get
long?
it.
Like, if you put that into the machine, how long does the test? Oh, well, does it take 4 hours period, or is it just like the prep that you're doing right now? The
So
prep.
the
Sorry, yeah, the
no, you're
prep that you're
good.
doing. No, well, I would just wanted to be on the screen. So, yeah, the prep that you're doing right now. Once it's prepped, is that what it's gonna like? The tubes, those are prep tubes. They're ready to be tested now.
So there's these here are already with certain chemicals in them. You just have to put the extracted DNA in here, so they're already prepared for you. If you try and like prepare your own like primer boxes or anything, you kind of have to know the chemistry in order to do it. But the prep part would be the DNA extraction. And with the DNA extraction, they put those away in the freezer. So this is my PCR. It extracts my DNA.
Okay.
Super hard to open, but it extracts my DNA for me. And especially if this is already being used at the minute, but this is my PCR machine and this is my qPCR machine. A qPCR machine gives like a real time results, so it gives you like right that instant results versus this gives you results.
Okay.
So this is more informative and actually takes the images and everything, and this kind of just extracts it all.
Okay.
Like on this here is where we put our samples in, and they're in these kind of like formatting. So I would take one of these out, and I can't take them out, but I would take one of these out and pipette everything inside of here. So all the DNA that I've extracted, it would be like um, it would be like this. After you extract the DNA and you put it in this, like here,
Right.
you get a sticker sheet and it goes over. And once you do that, you take this and you set it there, and you would take this and it kind of applies the pressure to it because this machine takes pictures of the infrared. Yeah, it takes picture of the different colors being expressed from the DNA. Sorry,
Okay.
I
Let's
stutter.
go. Let's go.
But it's like it has all the experiments that I've used prior. Like it would run the same experiment for me. So if I took this experiment and I wanted to run it, I can't really run it, but
Right.
if if I wanted to fit 96 samples in here, it would process everything. It drops down to a certain temperature and then rises really up to another temperature and stays steady and drops down. Because in certain parts of the DNA helix, the bonds will break at different temperatures. And that allows for those parts of the DNA that we're looking for to be extracted, multiplied by a million, and it shows present. And when it shows present, these are different cartridges that express different colors for different parts of the double helix or certain parts of the genome.
So are they like they're imaging cartridges basically? Like that's what
Pretty
they are?
much. So when you extract DNA, you're pretty much taking it. Well, with this one, with the qPCR, you're taking pictures of the colors being expressed from the certain um, genes that we're looking for. So then when it prints out the results or analyzes it, it'll show that these colors were expressed after reaction at this time or this time. If it doesn't express that color after the reaction, then it means that the pathogen or viroid is not present.
Okay. Okay.
Yeah. Um. So can I read that question?
Do we have any questions? Do you see
There's
one?
one. Yeah.
I mean, I
So
see, I see that. No, just
the
kidding. um, I see. I was totally zoned out on you. I wasn't even looking at the questions to be completely honest. Here we go.
That's okay. So whenever you're testing, I would completely, if you've been out in the field and say like you brought your samples in, maybe keep some scrubs in your testing areas so that you don't have any outside contaminants on your clothes, because that's the biggest part. If you don't have a whole like filtration system for your lab and you're just starting up, because there are a lot of machines like this that you can get cheaper and they kind of look like a USB, you plug them into your computer. But just always wear something that's completely contaminant free. Like I have to keep my plant material and the DNA testing separate just because it can um, put a virus there that I didn't know was there. So
Yeah, shout out to Bakashi Earthworks. You know, you already know, bro. It's coming. It's coming. It's very much coming soon. I like him to come on with me a lot because he's like, you know, if you know who he is, you've seen him on before, Brandon
yeah,
Rust. He's just so knowledgeable, man. I love to have him come through. So you already know, bro. It's coming. We almost at that couple week mark, man. I need you to come back soon. Yeah, I think she did explain it extremely well. And good evening, good evening, good evening. Hey, if you guys have any questions at all, feel free to ask.
yeah.
So I was curious, does it need to be filled? Like, do you need to have that full in order to run tests? Or that's just obviously what you don't need to. But it can only be used once. Each tray can only be used one time, or
Yeah, so whenever you extract DNA, you have to throw away whatever you use. Like,
okay.
because there's always a potential of leftover DNA in there. Even if you autoclave it, I wouldn't suggest it because DNA extraction should always be the most cleanliest thing that you do. Like, like OCD kind of clean.
I would be perfect.
Yeah.
I'm just saying.
Yeah.
All right, you guys, if you have any questions at all, you know, like honestly, ask any questions that you guys want. I know we're watching her,
Yeah.
but the questions are like they'll be on the screen where we can come back to the questions and ask. You had a question, TW. Let me look. TW. Wait, I think we just did that, didn't we? Yeah, I think we just. Pretty sure we just talked about that, didn't we?
Yeah. Yeah.
Yeah, that was the question I had up. Um, yeah, any questions you guys have? So, what was I gonna say? Okay, so that's basically that's basically what it is right there, right?
Yeah. That's pretty much all it is. It whole graph. Let me get this out real quick. But there's a big ass graph that comes out after that I just learned how to fucking read. So it took me a minute because I haven't done that math in forever. So
So you need to print this out, and you actually need to do math to figure out whether it's a positive or negative. Is that what you mean, or
so the graph that I got, um I was trying to make sure that it was all correct. And because the person that was supposed to train me on this, because I know most of it, but well, I'm not a pathologist. So
right?
but the person who was supposed to come out and show me didn't. So I got all the books that I could on it and kind of taught myself how to redo it again. But it'll give like in a graph, and in the graph you can see where the reaction happens inside of the tubes that allows for like the protein to kind of be expressed. Let me show you. All right, let me turn this around. Um, so over the next couple weeks, I will be testing a lot of strains. I think there's 180 strains that I'll be testing.
Mm. So are these tests coming from people that are well, obviously we should talk about that a little bit too, because she is actually run, you know, you guys can anybody that wants their clones tested, their garden tested at this point, she's ready to go. You know, last time we spoke about it in the will be coming soon phase. And as you can see, she's ready. So, you know, we'll talk about that a little bit too and see,
Yeah.
you know, price, or, you know, we could talk about how they can go about finding out pricing, or if you want to talk about pricing, what it costs, we could touch on the Mary stem again, and you know, what that takes to do, and also the male female testing. So
Yeah.
we'll talk about all that. All right. So what are we looking at here? What's the graph showing us?
So all these are the different colors from the different parts. Like these are a leaf, I believe. These two right here for one strain, and when it comes down like this and it goes up, this is just a reaction of the chemical being expressed, but it comes down. Oh shit! Wow. What the fuck happened?
Uh-oh.
All right.
Let's go
Hold on.
back.
Jesus. All right. Open. So you have to select your tests. So say if this whole thing was full, but you only wanted to check these samples here, you would just highlight the samples that you're requesting the software to analyze, and then you just click on so. But anything like this that is being expressed is simply just saying that the reaction happened inside of that sample, but it's showing a negative test. If it had, where's that one at? Like this one here, shit. That one could be a potential sample that actually has a viroid in it.
So where are you
Because
seeing that? Where is the difference? What was the difference
so
where you're seeing that?
shit. Like this one too, the way it goes here, that's the reaction. It plateaus and it goes back up, so that shows that it's slightly expressing in that sample that it's there. But it's not to a greater point. Like if it's past this 35 where it has the second hump, then that's when I would know that it's positive. And I didn't see any of the samples that express that.
Have you been finding test samples that are coming back positive? Like, what is the general, like, you know, I'm just gonna say percentage.
Yeah.
Like, say you're testing 50 plants, are you finding high rates of HLVD?
So since we just got everything, like I have all my TC material now, I have the I have everything. So that's why Monday's a go. On Monday, after I do the 180 samples, I'll be able to see, like, have a general idea um, of what a couple local farms have. And there's a couple smaller farms that are going to let me test for this to see if it's present there. But at the moment, I don't know. So starting Monday, I think Monday, Tuesday, and Wednesday, I'll be testing all day long and
Okay.
should try and get through as much strains as I can. And once I find a strain that is not expressing for my brain is shutting down. If it's not expressing any of the viroids, any of the other pathogens, because I think we have like 4 or 5 different testing that we do. Um, so if none of them are sick, then they'll move on to be put into the library for future production.
Okay.
And if they are sick, they'll be in quarantine, and there'll be some steps that I take to try and rejuvenate if the producer wants to move forward with it. If they don't, then they'll probably just be trashed.
All right. Well, I mean, we might as well. That was like the perfect lead into like the Mary stem, right? Because
Yeah.
that's what it is, right?
Yeah.
So,
So if if the material's clean, you don't necessarily have to take the meristem. For me, I take the meristem as a mother. That's my mother that I will take and put away into the genetic storage. But you can also take like little auxiliary buds and throw them in the TC and kind of promote the shoots to grow.
but you still so if you have something that tests positive,
Yeah.
you have to do it with the Mary stem, or you're saying you don't have to do it that way?
So if it's positive and the producer's like, yeah, I want to keep this cultivar or strain, then it has to be the meristem. And I would probably do 3 generations of a meristem before, um, and like let it grow out the full cycle, and then test the roots to make sure that it's completely clean. But it would probably take, that would take, depending on the genetics. If the genetics are really weak and really sick, then it might take a little bit longer. But so far I've seen, if it's just like a fusarium issue, it's not as bad. It takes about 6 generations of apicals to kind of get rid of.
What do we have? Okay. Well, I think we just sort of touched on that, but
After these next few weeks, I can post certain strains if the producers allow me. It just all depends on what they allow me to do, but I can always give you guys a general idea. Out of like 180 strains, like 30 came back with it. But I know that here on the West Coast, I've heard a lot of producers say that they're worried about that because it is going around, and it's harder to control because the hop latent viroid can be passed by insect. So if you have an insect that's not stationary and it just bites your plant and has a swap of sap exchange and goes to the next plant, and it's a carrier, it can it can just go into your garden and pretty much just spread it.
um, okay. So I mean, we could talk about, this sort of can go into the male the male female testing as well.
Yep.
Have you seen any traits in leaves after testing to male or female? Or any traits in leaves
So
after testing?
do
Okay.
you mean like testing for the hop latent? Because if you have that, you can totally tell from the leaves. Um, Sometimes the leaves have this weird kind of fringe look to it, not like a cool mutation, but an actual. You can tell it's a symptom or a sign that there's something being expressed. I can post some pictures of what it looks like.
So when you're seeing plants that are positive, is there always visible signs, or because it's latent? Um, do you even always see, if like there's a cultivar that might have it, but it's latent, as you say, pop latent virus, like is there times where it will grow all the way to term, all the way till it's ready to harvest and not see it, or will it always eventually, show some kinds of visible signs?
So I would say it all depends on the strain, its vigor, like if it's a strong plant, and the genetics. Because sometimes a plant can have a hop latent viroid and you won't even tell. It won't be stunted. It still throws a high yields. So there's not always expression of the viroid being present, but a majority of the time it does show once it reaches maturity, and you can see it. It's either stunted compared to the plants around it, and sometimes the buds are a lot more arid. You can tell from the structure of the plant of the cola that there's something going on. But there's a few different signs that you can actually see it, but there are some cultivars, and those are the ones I really want to study more than anything. I want to look at the genetics and see their genetics versus the sister genetics or twin sister genetics to see exactly what the resistant one has and the non-resistant one has, just to kind of compare. But
Has there yet to be found a strain that is totally like, has any, is there any strains that they've tested or you've tested trying to maybe introduce the virus, and there's, that they're immune to it, so to say, for lack of a better
so
word.
I have a producer that let me have a sample of one of their cultivars, and I purposely infected it to see if it would like mutate, what exactly would happen. And it didn't express anything for quite some time, but I think like once it hit flower, you can totally see the nugs were not really formed. It just looked like just calyxes, a bunch of them just spotted on the stem.
Right.
But that's just one plant. If I had the ability to test one cultivar and like 50 different plants because every plant is different.
Right.
It would allow for me to see, but I once I get a bigger testing facility, that's the hop latent viroid is something I've always loved to to study, and I've been studying that thing for like 4 years.
So, okay, the hop latent viroid, I
Yeah.
mean, like some of the back or the history of it, right? Hops. So is that
Yeah.
like, and then like, do we know when, how it jumped to cannabis? Is that known, or is that
So that I'm not sure, but hops is cannabis cousin.
right?
So if the host has similarities that one pathogen likes, it can move on to another one. A lot of plants, like pathogens, they find a host that's comfortable environment for them and allows for growth to happen. And if it has that growth, and it's not all plants, it's just a host plant that's similar to the host plant. So, I feel like since hops is so similar to cannabis, it allowed it to, especially with aphids, because aphids are a big problem. So are mites. Mites are a really big problem in hops. So there is a possibility that, maybe someone had something on and insect was on there and they went somewhere.
Right.
But that's the only way I could possibly see it happen, just because of the fluid exchange.
Okay. Interesting. Yeah.
But I'll have to get back to you on that. I don't know the history of that. I just read a lot of papers. I think it'd be interesting though, because it kind of maybe would
Right.
allow for better understanding.
Yeah. I've always been curious about that. When in time, when was the first cannabis plant? Like, when did we find it? What year? How long? Just
Yeah.
curious about those things too.
Well, I know over the last few 5 to 6 years, more producers have become increasingly worried about it because
Mhm.
it's more in like California and Washington that a lot of them have a lot more issues versus here in Oregon. At least I haven't heard so many, but I feel since there's so much plants that are not being tested and being sold, that it allows for quicker spread.
Yeah, and I feel like that's like a huge thing, right? Like, I feel like if you guys are buying clones and it's not tested yet,
Yeah,
y'all should make sure that's tested. And I feel like like to all the clone distributors out there, they should all be tested. Like, before that stuff's even sold, when they
yeah.
get it in, it should all be tested. Every fucking plant at this point
Yeah.
should be tested. So
Yeah, especially if it's starting to affect a lot of people. The industry's kind of already struggling, especially here in Oregon a little bit. So, and I'm sure most places because the pounds have went down in price. So, if a
right.
producer's struggling in more than one way, we should at least help each other in the community to kind of lift at least that a little bit off.
Yeah, no doubt. Um, okay, so all right, this is going back to his question. That was my concern, where leftover residue or whatever could house so many bad readings. So I was wondering how you keep the work area so clean. And I think we touched on the alcohol.
Yeah.
Um,
I like when it comes to the lab, I am very, very particular on the cleaning regimen and what gets brought in here. Hopefully, eventually soon, I'll have my own desk area so there's no food because technically in the lab there should be no food or water because all that starts to evaporate into the air. It could be a possible contamination. But I sweep. I have a little robot that sweeps for me and mops for me while I'm gone. And then
let's go.
twice a month I get down on my knees, I pull everything down off the shelves and I clean everything from top to bottom, and then I use DI water after I use the bleach, and then I clean it again with the DI water, and then after I let it settle after lunch I come through and ISO everything, just so it smells like straight chemicals in here,
Yeah,
but
it's a
it's
lab, man.
it's
It's a
clean,
lab.
man. It's
Yeah,
super
it's expected.
yeah.
Um, all right, I seen somewhere today about prices ranging per unit to per test. So is that? I mean,
So we buy our chemicals. already prepared, so I don't have to do that whole chemistry thing I talked about. I love chemistry, but not to the point to crying to try and get all these primers, buffers done. Um, but we buy all ours from a company where we bought the machine, and um, they're pretty expensive, but still it's beneficial. Um, for like single testing for just one is like $40, and I know most places are $45 to $50 per plant to be tested, but I think here it'd be 40 for now.
Okay.
Unless, and if it's like a really big sample, then I think that would be something I discuss with like my boss and like, hey, because at
Right.
the end of the day, that's still my business partner, and.
Right. All right. So Bokashi, back up. I set up a tissue culture lab. I'll also be doing marker assisted breeding.
Hell
So
yeah.
let's I mean I like next how about next week we talk about the marker assisted breeding. Brandon, next week we could talk about that a little bit. All right, I'm gonna take that as a yes, bro. I'll be thinking about let's see. Okay. It's bad. HLBD is everywhere. I test my plants a few times per year.
That's cool.
COG7 mutation is a gene that shows HL. Oh, there you go. So is that's sort of what we were talking about. I know I'm a little bit back on the comments, so
Yeah.
but good info. You're spot on.
Thank you.
The latent part is also in testing. Sometimes it doesn't show in. That's what that's why I feel like the latent part of it, like it's late. It is exactly in the name. It's latent. So, and that's why I was asking about plants growing out the term,
Yeah.
and you not really being able to see it, or if there was always visible signs.
Yeah.
What are some of the visible signs for people that might be in their gardens and don't know what to look for?
So if your genetics are pretty on point and there's a lot of uniform to your genetics, but you see one of your plants that are just a lot more stunted than the other ones, and then if you keep on producing and it's still like stunted, um, that's like always the first sign is usually the stunting of your plant, and the second is the flower. The yields are way lower than the plants usually around it.
This is incredible. I learned more of the sciences involved in growing, not just following a protocol without really knowing why you're using all these chemicals. I want to go full organic. Brandon, let's go. Yeah, almost like an operating room or a clean room.
Yeah.
Okay, so are you familiar with the COG7 mutation?
So I know I was talking to someone about it, but for right now, I'm trying to work on things that people with a typical garden have access to. So the things when I'm experimenting with everything, because a viroid can't be expressed like mold and everything else. So I really don't I really didn't get my machines until now. So yeah.
When did you get them? Just like within the last week or so?
No, so these machines aren't sequencers. There's different machines. This machine is just a qPCR and it detects whatever I like the protocol that I put inside of it. So whatever I put in it, it'll amplify that. But I don't have a sequencer. I wish I did, but no. That's next.
Okay, so listen, I know I say this a lot during the live, and I'm going to say to Trey K. Trey, I see you over on the gram, on Instagram. You see that at the bottom right there? I
Yeah.
see your questions. That's why I'm looking down. I'm trying to keep up with the questions over there. But you guys on Instagram, there's a bunch of you guys over there. Just come over to the YouTube so I can throw your guys' questions up on the screen.
Oh, yeah.
As you can tell, it's a different setup. Like you're seeing us on Instagram, and we're really on YouTube, Twitch. We're on a couple different platforms. So it's hard for me to keep looking down. I have no problem doing it, but it just makes it so much easier if you guys come to the YouTube. It's really easy. It's one word, Dark Star Genetic Feedback. And then you could ask your questions. I could throw them up on the screen. But in the meanwhile, Trey K, My friend says, I have a plan from Steve that is sus. Mine is horizontal branchings that people alerting me could be HLVD. So that was what she said.
Huh. I'd like to look at it.
There you go. Okay, so what do you think on do lamprey strains get HLVD?
If it's cannabis, it's a plant. So, yeah.
Yeah,
Uh,
yeah. So, like she said, like, think about it in the terms of like us as human beings.
yeah.
This is a virus, so it doesn't. Cannabis is cannabis, like we're human beings. So just look at it like that. So yes, yes. Let's see.
That's cool that they can do it for 25.
Had to let yield goes down 20. Okay, so small grow might not tell. Shout out to Tino. What's up, my brother? What up, Doug? What up, 808? Shout out to you. I'm gonna leave it up there just for 1 second, man. It's always Tino's fault. Just know that. No matter what it is, it doesn't matter. You might spill your drink. You might drop something on the ground. The phone might not be where you thought it was. Tino's fault. All right, just Tino's fault, guys. Moving on. Moving on. Let's go. I like this guy. This guy, nice, loving these topics, so educational. I want to thank everyone involved for all the dedication. Absolutely, man. Our pleasure.
Yeah.
Yeah, it's Trey K. We see her over there. We see her. Oh, he's talking about. Okay, so are you talking about HLVD testing, or are you talking about, that sounds more on the lines of like male female sex. Well, I guess that would be more expensive for that, but.
So, when I test for the 4 primary in this state. Um, the fusarium, the viroids. I think there's like 4 different viroids that I test for, not just HPV or Hoplayton. And then I can't remember the last one, but I think it's like 7 different pathogens and viroids that we test for. It's not just one, unless you just want that one that's different. But we try and make sure that anything that's being bred or moving forward to be placed in the genetic library, that it's solid. Like there's nothing that would risk its genetics.
Yeah, I mean, I know the other companies are right around there too. So that testing to me sounds a little. Can I speak like the kids speak? Sus. Sounds a little sus. Just saying, it just does.
Yeah.
I work. I mean, I have stopped working with them because I'm probably going to send people to her. But I work with Farmer Freeman. I got another guy Tino knows. We try to get some things together with him, and they're all around that same price. So yeah. That $25 sounds sus. Let's go. All right. So in certain environmental conditions, question is, so can certain environmental conditions bring a mutation out? And see, this is why we need Max up in here. Where you at, Max? This is what I'm talking about, man. Yo, where's Max at? We're supposed to have somebody else on with us tonight that is actually the perfect person to ask about mutations,
Yeah.
because he just is. So let me see. Where's Max at, man?
Yeah, me and Max, man. I printed out so many papers on mutations for him because that's his jam right there. I just like looking at them and looking at them on the screen, but
Hey,
yeah.
Tino, are you still in here? Hey, can you give Max a call? I just can't get off the other phone to call him. If you wouldn't mind, if you can, it's cool. I'll stop. I'll figure out a way to call him. But if you're in here and you can hit up Max and let him know we're live, and maybe to come over here. I sent him an invite on the 6 page too. If you could. If not, it's cool. I get, it's cool if not. What else do we got? Okay, is it more prone? Is it more prone to affect strains that are inbred? Is it more prone to affect strains that are inbred? More inbred.
So
Oh wait.
I would just think if the strain has weak genetics and then it has that stress, the stress is more of it reduces its like vigor, its ability to be strong and fight against things. So if a plant's already weak in its genetics and it's stressed out because of like whatever the environmental stress is, or just because you accidentally dropped a plant and kind of broke a piece and there's an opening, it just all depends on the situation and the environment, because the environment has a lot to do, and how you like if you use scissors to cut, are you dipping it in or wiping it with ISO as well as bleach? Are you just cutting it from plant to plant? It just kind of depends on a lot of different factors.
Hold on, I'm trying to get us back. They cut my lives back down to an hour on this page, so I'm just trying to get us back on Instagram real quick.
Okay.
Give me one sec. Here we go. Oh, hold on. Go. Yeah, I hate that happened. Instagram is such a pain in my ass, man.
Yeah, I'm blocked from lives until November 1st, and it's like really hurting me.
You know, I mean, I'm really grateful to have this platform right here to be like really. Yeah, Instagram just sucks. It just sucks, man. It just does. You know. All right, we're back on. I think we're back on now. Hold on. All right, we're good. So 2 joints. I know who you are. I can't change my YouTube. Oh, okay. Oh, I know who you are. Okay, I didn't know who it was at first. What company has been testing positive? Yeah. She just started to use them to start to do the test now. So, wait, do you test the roots? So, yeah,
Yeah.
that's exactly what we talked about when we first started, and that's exactly what she does test. Yes. You just did. I just tried to when the Instagram stopped. He didn't answer. So, who knows? I wanted that Max on, but we'll see. We'll see if he shows up or not. You know. 808, they took down your instance again. We already know. Tina one is everywhere. I'm not putting that up there. I was sick. What if I'm in Canada? Can we ship for testing?
So at the moment, I don't do anything internationally, just because I'm a mom and the way federal laws sit. I need to make sure that I watch my P's and Q's for right now. Um, but I'm sure there are places in Canada that are reliable that actually test for that. And if not, I could try and search for some and see what I could find.
Yes.
Yeah.
I didn't. I just clicked on it by accident. I don't think she said. I think that you're confused, sir. I don't think she ever said anything about outdoor weed sucking. I think you're just confused. Yeah. You must be from Canada. Sorry, but not
To me,
sorry.
the weed all depends on who's growing it and how much they love and know what they're doing.
How can you tell if the you could touch on that? I know we already touched on it a little bit. So
Um, so without testing, like I said, if you have a lot of like with if you have more than one plant in that cultivar, you're looking down your row or in your room or wherever you have your plants, and you can see the difference that's completely stunted versus the other ones, but the other plants are more uniform like, then I would probably say, pay close attention, but maybe pull that plant to the side and put it in quarantine just in case if it were to have it, you wouldn't want it to spread to your other ones.
I just saw another. Wait, what did I just say? Oh, I mean, here's interesting question. Really,
Yeah.
is HLBD harmful to us humans?
I don't think so. It's more affects the plant and lowers the yield. So I mean, if you're a producer, yeah, it affects your pocket, but um, I I don't feel that it affects it in any other way, like humans.
Uh, is there more? Is there more? I guess is there more prevalence of HLBD geographically?
So I haven't heard too much of it on like the East Coast. I know there's some people that have been asking me questions about it, and like kind of how to look for it. And but it's I have a lot of producers, especially here in Oregon, that have been asking me. I have a few in Cali and quite a bit in Washington as well. They're like, hey, they're telling me the signs and symptoms, and so I feel for right now, maybe it's in this area. I'm not sure exactly why. Maybe because there's more production outside, or I have no idea. Maybe people get clones from the West. I don't know.
Okay. Do you think bleach affects the biology of the cuttings?
So if when you're doing tissue culture, I feel like depending on what part of the plant organ you're extracting and what method that you're trying to use or approach, um, I would say think of the pressure inside of the cell and outside of the cell and how it affects with chemicals. So some chemicals have charges that might affect the way the cell works and some might not. So maybe when you're ready to do a certain method or approach in tissue culture, maybe consider the concentrations and the impact of the cell.
I mean, you know, they know, they know. Canada, man, don't get me started on Canada. Just don't get me started. Uh, we're losing our accounts. What up? Shout out in the house. Let's go. Mm-hmm. So we have, this has been such an informative live. Thank you for sharing the process with us from Instagram. Come over to YouTube so I can put it up on the screen. um, let's see. So, okay. So Inhouse said no on that. She just explained. Yeah. I already know the answer. All right. They found it because big company yields were 20 to 30% lower for no reason on a small grow. Hard to tell. Think you're just, it's hard to tell. You're just think you messed up the test. It's hard to tell because you're not sure because it's such a small grow. So, yeah, I mean, I would assume this is what the question,
Yeah.
I was curious. Exactly. So in-house, I'm going to put that up in 1 second, but I'm curious if Brandon, like when do we know when there was the jump from hops over to, well, I'll put that up because we were already talking about hops before you came in probably. And, we were curious a little bit. I was curious if we knew about when or can narrow it down or pinpoint it to when, it did the jump over from hops to cannabis. I mean, it's curious.
Yeah.
It came from hops past to EPM. What? Wait, I have no idea what you mean, bro. I don't know what you mean. It came from hops past to when they roast them like dummies. Spelling, please. Can you put on the spell check? Can you hit the spell check button, bro? I'm just kidding. All right. Yeah, it's from hops. We already talked. Yeah, we talked about this already. So, yeah. 1987. Okay. No, I know it's nothing new. I just didn't know when it went from, you know, if we knew when it sort of made that jump from hops to cannabis. That's all.
Yeah.
All right. Let me see. Christian chat. Let's go. I think it came from Hip. It's just so stupid, bro. When they crossed, is that when it happened? So this, okay. This is, you know, like I said, I was curious. I mean, if that's really, so we're talking about 2014, so you're not sure? Okay, interesting. Oh shit, let's go. I mean, I'm just saying. Finally, reiterate. Thank you, Ade, for sharing the information with us all. You definitely educated. Yeah, let's go.
Oh, yeah.
Perfect. That's why we do this.
Definitely.
And I'm glad you finally found the YouTube too. I'm just saying, bro. I'm just saying. It was like Ross. I'm like, what? Oh shit.
So
I don't expect any less at all. So we good. You know, we're all on the same page here. Okay, let's see. It's probably been around decades, but plants are in constant state of health decline. It shows predominantly. Okay. There's plenty of research from the late 80s and 90s.
yeah.
Just go to look up when I crossed them. It was done in the lab. So, okay, so at least, you know, I guess it's when they crossed the hops for him is when it happened. Interesting.
I had so much fun being on your live and talking about everything. I appreciate that.
I'm just going to leave that one. I'm going to leave that one. I'm not going to put that up on the screen, but I hear you. I got you. All right. I just can't put it up on the screen from Instagram is the problem, you know? That's where the problem is. And it sucks. I wish I could. Like, I don't want to have to look down at the phone. I'd much rather just be able to look here. So, well, I'm glad that you made it over here with the full name. All right, so I came in late. I don't know if you discussed it, but if you have a plant that won't clone easily, is it a likely candidate for HLVD? Not always, but definitely very. Wait, are you answering your own question or?
I know something won't clone easy. It's probably something in genetics, but I don't know so much if it's, you know.
I believe the virus drew to inbreeding like HIV. Too many people breeding for the next big. I don't think that's what it is. I think we sort of just went over what it was and how it happened. To be honest, I mean, I do want to talk about male-female testing a little bit. I'm just trying to get through the questions. Those who discover are the ones who provide the solution. That's exactly. Hey, man, that's like what it is, right? In-house, that's what it is. Yeah. This is what they do. They create a problem to create a solution, and that's what happens. Oh, let's go. Barebone, that's the guy I was just with down and let's go. What's up, bro? I didn't know it was you. Let's go. I'm
gonna have to hop out of here. I gotta go take the kids to parent-teacher conference night.
Okay.
So, I appreciate coming on, and
You got it.
I'll post the results probably tomorrow in the afternoon.
I appreciate you coming on. I'm definitely gonna have you come back. I know I kept you longer than expected. I expected Max to be here. You know, I really did.
That's it, man. We gotta tell him no soup, right? Like you say.
No soup for you. Yo, will you do me a favor though? Can you hit him up on the phone? See if he answers and just tell him, you know, because I have about 40 minutes in between now and the next guest. So if you wouldn't mind, just shoot him a call and just tell him if he wants to jump on. I'm here.
For sure, for sure.
All right.
Oh yeah, have a good one.
All right, I appreciate it.
Bye.
Bye. Bye. Damn, all alone now. Hey, in-house, you want to jump on? You busy? And I got about 40 minutes to kill, so, you know, we could do a couple giveaways. I'm not going to have these other guys on until 10 o'clock. That's 40 minutes. 40 minutes, 40 minutes, 40 minutes. All right. If anybody wants to come in, just let me know. I'll send you a link. You can come in. Tino, in-house. Anybody that wants to come in, let me know. Otherwise, we're going to sit here and kill about 40 minutes. You'll jump on? All right, perfect. Here. I'm going to send you a link. Hold on. All right, I sent you a link to the rec email. I think it's the same place I sent you the link last time. So, you know, the rec email is the one I sent it to. Well, it's Tino, but yeah, it's always Tino's fault, man. I'm glad you're on board with that, you know, because it's always just is what it is, man. No matter what it is, it's fucking Tino. Yeah, I don't know who Gino is, bro. I just said the same shit. I'm not sure who that is, but you know, let me make sure in-house got that. Let's go. Let's go. It's always fucking Tino's fault. I'm just saying, bro. Like, I just literally dropped something, you know, right here. It just happened. Just happened. Fucking Tino's fault. I'm telling you, man. I'm telling you. Tino's fault. Gino's Pizza. Hey, did you get the link? Let me know. We'll do a giveaway in the meanwhile. How about we do a giveaway? Let's go. Let me see if I have the giveaway tool up. All right, let's do this. Here, let me get this up here. We'll do a little giveaway. Not for you. No soup. If you're on Instagram, you out of gas, man. I'm just letting y'all know right now. It's not going to work. It's not going to work. Hold on. That is from the last one. Let me redo this. Hold on. Oh, wait. Hold on. Slow down. All right. Yo, we might as well use that as the hashtag, right? Let's do that. Let's make that the hashtag. Y'all ready? Let's do it. Here we go. This is the hashtag. This is for the giveaway. Bam. Let's go. Let's go. Let's go. And go. Go. That's the hashtag, y'all. Go for it. So all you got to do is type the hashtag in. We'll pull up the thing. It'll pull a winner, and somebody's going to win something right now, and we'll kill some time. So I just said in-house over the email. He should be jumping in here in a second. So try to keep him until 10 o'clock, and then we'll have our glass blowing, community talk. Yeah, it's going to be fun. So let's go. I'm going to leave it up. Wait, let me go back to the original one, right? We might as well leave that up on the damn screen. Where did it go? Oh, shit, I lost it. Damn, I told. Oh, there it is right there. Well, always Tino's fault. Always fucking. Let me turn this. Let me turn the camera off. Give me 1 second, man. Hey, Tonic, she's gone, brother. She been gone a couple minutes now, man. So, yeah, she gone. She gone. She had to take off, bro. Let's make sure he got that. Man, I'm just chilling. I'm actually trying to make sure in-house got the right link sent over to him. That's actually what I'm doing. It's fucking always Mike's fault. All right, here he is. Let's go. Ugh.
What up?
What is that? What is that? What is that?
Oh, you know, just I gotta go get a haircut, so I'm gonna get this mop chopped off.
I mean, you know, it looked pretty good though, bro. You going to get it chopped off for real? Yeah.
I'm gonna go get a haircut. That's what I'm on my way to now. But I'll jump in here. What's going down with you?
Not much. I figured we put up the Tino's fault hashtag for a giveaway. I'm gonna take it. You know, we're gonna do a giveaway. We're gonna spread the hashtag around a little bit because we already know it's always Tino's fault.
So I've been remodeling my house and I banged up my hand, right? So I like pinched it. First thing I thought, God damn it, Tino's fault.
Exactly, man. There was like
Everything.
10, 20. So many things happened today, and every time it was Tino's fault. I mean, it's always Tino's fault. Yeah.
Yeah. The days are getting shorter because we're heading into fall. Tino's fault.
Tino's fault.
But yeah, you guys had a pretty good discussion going on there. There's a lot there.
A lot there. I love to have her come back, and I will. I didn't know she, you know, I only meant to keep her an hour, so she did stay a little bit longer than expected. But those are good. You know, I really love to bring people on like that. Like the next guest also, well, something totally different, but they're going to be blowing glass, talking about how they, you know, it's cool to just be able to bring all these different parts, aspects of the community together. I really love to have her come on just because she has so much knowledge. And I thought, you know, Brandon Russ was in the comments, having you in here as well. It just, you know.
Well, you can also see, you've had her on a few times, and you can tell that she's really advancing and everything too, because especially more knowledgeable every time she comes on.
Exactly. And it's really nice to see her, you know, her progression. It just is. And I talk to her a lot. Like, she'll message me a lot. She'll ask me questions. I'm always trying to help her out in any way that I can. Not that I really can, but, you know, just the conversations I have with her, it's cool. So I've really been along for the ride with her, as well as her, you know, kind of research Max. They're really good friends. He helps her out with a lot. I didn't even know that at first. I had no idea that they knew each other. So they're all apparently in, you know, Tino's area, I would guess, because Max, I know, and Tino are close. So yeah, small community, but not.
Yeah, Tino and him bumped into each other at the grocery store. It's funny because that's one thing I kind of miss with all the grocery stores going away, is the people that over the years that you just bump into by going into it, you know.
Absolutely.
So you kind of like point at each other, and you're like, and then you're like, yeah, I'm that person, yeah. So,
All right, this
so
is our winner right here. Let me pull that down. So if you're here, just comment. I'll write you down, and then you'll hit me up after this live. You can either email me, or if you're on Instagram, hit me up on Instagram. But let me know you're here. There he is, right there. You're on Twitch. All right. So you can email me. Here, let me just put this in. I didn't know how long it was going to take for you to come on. I figured we'd do a giveaway to waste some time. So shoot me an email at that email right there, buddy, because I don't know who you are. So I'll put it up on the screen for a second. Take that email, shoot me an email, and we'll get you covered on the giveaway. All right, let me take this back down. All right, there we go.
you know how you remember back in the day, you come up behind somebody and you just kind of take your toe and put it into the back of their shin, not their shin, but their kneecap, and it kind of like dip down. You ever done that to somebody?
Yeah, of course.
I did that a few weeks ago. I was standing in line at the post office, and Jinx Proof was about 6 or 7 people ahead of me. And he's distinctive because it's an orange hat, right? So I kind of just snuck up behind him and did that to him.
That's funny.
Yeah. No, apparently we live in the same neighborhood.
Damn, I seen him come on with you before one time. I know you've been in lives together.
He's got he's got a glass shop right down from one of our places, and so we're all like in basically kind of the same vicinity, same area. But that's what I was saying is like that's why I miss the grow stores, because over the years you go in there and then you never knew who you were going to bump into, and then also people that you met and watch them kind of blow up in the process, too.
Yeah. Man, it's crazy. I live in a major city. There was one grow store, and that grow store is gone. And it's just sort of, it blows my mind. I mean, I guess it's because, you can't really grow, but still, it's just.
Well, we
like
come from a time where you could throw a rock and you pretty much you could throw a rock and you would hit a grow house of some sort. And then, so there was quite a few of them at one point. And then, but as obviously medical took place, and then eventually recreational, it just kind of slowly died out. Everybody moved to east, actually.
They just seem like they're becoming like a lost, there's just not that many of them. I mean, there's like 1 or 2 in Jersey. In this whole area, there's one really. That's it. Well, I mean, because you have Amazon, you have like all these other ways to get stuff sent to you and stuff. I feel like it's sort of crazy. So
well, if you're gonna blow out a couple of houses, wouldn't you rather do it closer to your market than to try to take it from the West Coast and then take it all the way to the East Coast when you can just go easily set up over there?
Yeah, absolutely. Yeah, no doubt about that. Yep.
Yeah.
So what's up?
I'll
What
tell
are we
you,
doing?
they used to do some crazy stuff here, though. They used to tap right into the main line, so they would dig down to the main line and then stick pennies in it, and then jumpers onto that. That's about as dangerous as you can get if you're going to tap into the main power line.
Yeah, but I don't fuck with electric because I've been electrocuted before. So, doing shit like that, like I will always have an electrician at this point. But back then, yeah.
Always respect. Well, that's the thing is back in the day you had to learn calc load, you had to learn all that, because it's not like you were going to hire an electrician, and you definitely didn't. Everybody that came into that knew about your grow is a potential witness. So that's where you're like, all right, now I gotta learn a lot of this myself. So that's how you, especially electrical. Electrical is dicey, and it's actually the 120 is more dangerous than the 220, because 220 will basically buck you. 120 grabs a hold of you and holds you tightly.
Yeah, you can't it's fucking dangerous. It just is. But you gotta learn everything. Framing, to build your own rooms, plumbing, everything. Everything back then was difficult. You know.
Well, the construction part was pretty easy for me because that's my background. So,
Yeah.
Framing, building, all that. But learning learning plumbing or being able to teach myself how to move hundreds of gallons in just a few minutes, just with the flood tables and all that, and we had a couple of 500-gallon tanks. So you wanted to pump that water as fast as it could because you didn't want it exposed to light for too long. Because if it's exposed to light and it's got nutrients in it, it's going to form algae.
Yep.
So you want to pump that in and pump it back out as fast as it went in. You want it to come right back out.
What's in-house working on new these days?
Well, first I'm trying to catch up on my sleep, which is not really working out. But that's just every time I travel, I get all twisted up because then it's like your sleeping patterns are off and everything, and then we're losing like some daylight. But as far as like
You
breeding
know whose fault
projects,
that is, right?
what's that?
You know whose fault that is. The
Oh,
losing
that's
daylight
100%.
part.
Yeah, Tino's fault. Yeah, no, I forgot to. Well, I think by this time everybody knows that. But yeah, but yeah, no, we did. We just wrapped up a few of these blue unicorn crosses, and then did kind of some of these few other crosses that are kind of out there. So I'm gonna pop those and kind of see what comes out of that. See if there's anything good that comes out of it. But also, kind of it's not so busy right around this time because we got the outdoor that's coming up, and then starting to get everything reset for next year and next season. So this is about the time when it's like kind of the calm before the storm, before it all starts getting crazy again.
Yeah, harvest is coming soon. Yeah. Well, a lot of
Yes.
places. Yep.
You know what I'm really happy is all the places. It's not. We had a little bit of smoke this year, but not just days and days and days of just covered in smoke. So we should have everything should go great this season.
Tino's fault. Smoke. Tino's fault. Canada's fault. Tino's fault.
Actually, Canada didn't burn down as much as it normally does. Normally, every year for the past like 5-6 years, Canada just stays on fire, like for like 4 or 5 months out of the year.
Yeah, I've been trying to tell him, man. We want to build like a wall and keep that shit on your side, you know, just to make sure from now on.
I gotta be careful when I talk about Canada. I made a lot of Canadians mad yesterday.
So. I always make him mad, but you know what? They know I'm just playing, man. Except the one dude, bro. And then this morning, I had it. Yeah, hold on. After our live, I ended up with Canadian Bacon Mike, and this shit was funny. It was funny. I'm just gonna.
They said, "How dare you come after our fearless leader Trudeau? You don't know nothing."
Oh my God! So what are we doing, man? What are we doing tonight? You know, are we gonna revisit last night after this?
We'll probably just kick off the live and see what pops up. I guess apparently it ruffled some feathers, but I don't know. We'll do something. We'll do something fun.
All right, cool. Yeah, definitely ruffled some feathers. You know.
Well, yeah, it's yeah. Well, I finally got the lives back, so we had a lot of fun yesterday. A lot of people were having fun in the live yesterday, so see if we can keep it going.
Yeah, man, we went 3 hours. You know, that was a good time. That was.
If it sucks though, then if it sucks, it's Tino's fault.
I mean, yeah, of course. Whose else's fault would it be? Not ours. It's definitely Tino's fault.
Yeah. But I want to start planning. I want to get over to Europe next spring going into summer. I want to go try to see if we can spend 2 or 3 weeks over there. I mean, it's going to be that's a long time to get away, but I think it'll be well worth it.
Are you thinking of Spain at all, or just like where in Europe?
Well, everything in Europe is relatively pretty close to each other. It's not like here in the United States where if you want to go somewhere, it's like everything's so far apart. Like, especially you guys have it good on the East Coast because everything's like nice and compact and tight there. But on the West Coast, like we want to go to the next major city, it's like 3 hours away, 4 hours away, on a drive or 2 hour flight. But yeah, in Europe, everything's relatively close and they're pretty connected by like train and everything else. So, but it'd be nice to get over there.
Yeah, the Autobahn or the train. Yeah, the one train. I can't remember what it was called. Right, that connects pretty much all your. What's it called? The fucking train.
Oh, if we went to Germany, I'm definitely getting down on the Autobahn.
Oh, definitely, man. Yeah. I mean,
so the cool part is, a friend of mine works for Porsche, so I'm pretty positive probably get a vehicle that can be fun to drive on the Autobahn.
Oh, look who just showed up. Let's go.
Yo.
All right, Canna Research in the house. All right, the filler is over. You guys, the show can begin.
Go get your hair shaved off, bro. We'll see you in a little bit.
All right,
man.
I'm gonna get perm.
Oh, yo, he got in the perm. Y'all make sure you're gonna tune in tonight. Should be interesting. All right, Tino's fine.
All right, I'll see you.
All right, later. All right, cool. So we actually have about 20 minutes left until these other guys are coming in, but yeah, I was trying to get in touch with you earlier. Addie's pretty much gone already. But we were talking about, there were some questions about mutants and stuff, and man, I wish I could remember what the questions were or find the questions, because you're obviously the perfect person for that. So let's see how long we can go before they get in here. If you want to talk, introduce yourself a little bit for anybody that doesn't know who you are.
Yeah, I was able to hop on the laptop this time with this. On the lives, I can't do that, so it's pretty cool.
So much better, right? So much better on here. We're actually on Instagram, Facebook, Twitch, the whole nine. You got to bring on 808. Yeah, I don't know. You have to hit him up on that one. But all right. So yeah, introduce yourself. Let them know who you are, and we'll see how long we can go. I tried to call you earlier. I wanted to have a whole hour. I wanted to have you guys both on together. Really, is what I wanted.
Yeah, I was trying to, bro. I freaking picked up the girls from school, took some edibles, and just knocked out, bro. I'm still having a kind of a edible.
No, don't blame the edibles, bro. Blame Tino. It's always Tino's fault. That's all. It doesn't matter. All you gotta do is say, "Hey, man, it was Tino's fault," and I'll be like, "All right, cool. I know."
I'm on a little health kick, bro. So I haven't really blazed up for a couple weeks. Just all edibles, and my lungs are feeling better. Feeling good.
Questions coming in. So, yeah, Canna Research is who this is, guys. Specializes in mutants, like the Freak Show. The Freak Show. He could tell you a little bit about that. The Berry Freak. I really wish we had more time. I don't want to rush through before these guys get in here. But let's talk about as much as we can. Maybe
they're asking. I have a question. So, yes. I mean, okay. So, essentially, you have. I think what they're asking is, when plants are coming out with 3 leaves around from the center rather than the typical 2 on each side. So that's a mutation that you see sometimes. And yeah, it could go up to 4s. It could even go up to 5s. So that's called the phyllotaxy. So the arrangement of the leaves around, or the nodes around the center of the plant that's coming out. So that's what's called a world phyllotaxy. So your typical one is alternating, where you have one leaf coming out here, one coming out here, and occasionally you have one here, and another one right there. So that would be essentially, a 3 times phyllotaxy, and then if you had 4, it'd be a quadruple phyllotaxy, and that's just the arrangement of the leaves on the stem. And it's something that people have bred towards before, and it's not it's not consistent enough in the germline to be something that you're going to see a whole line of quad world phyllotaxy strains. Like there's some that I'll see more of it. I'll see occasionally in some of the berry-free crosses a quad world phyllotaxy. I even had some actually that came out of the watermelon and later that made the watermelon Rolex. There were some that were quads. And it's something, you could select for it or against it. But when you select for it, it's not necessarily going to be something that could be, consistent throughout the offspring through the prodigy. But yeah, I mean, that's something that you could also try to clone it, and it usually goes away. You'll see it go maybe like 4 or 5 sets of leaves, with a world phyllotaxy, and then it'll go back normal. And I don't know. The premises behind it was people thought, you could get a higher yielding plant because you have 3 points of flower that are coming rather than 2 or 4, But it's not been something that I've, been too crazy over trying to select for, because from what I saw is once you cross those plants out, it doesn't necessarily come out in the offspring, more.
We have. Okay, so like I just want you guys to all know, like any questions you guys have about, like he really is. You could ask this guy anything. Like he has, he really is so knowledgeable, man. We sit and talk a lot, and we'll sit on the phone for hours. So, if you guys have any questions at all, especially about like mutations, mutants, berry freak, freak show, berry freak, all that stuff you guys know about or heard about, this is the guy you want to ask these questions to. I'm going to try to get through as many questions in a little bit of time that we have, and I really am going to force him to come back next week for a full hour, I think. But yeah, I'm going to, bro. Because I just feel like, like our conversations, like we literally sit on the phone for hours, really, and they go by like that. And you just do have so much knowledge, man. Like you are just one of those, you've just been doing it your whole life, or fucking most of your life. Second generation, second generation,
Second generation breeder, yeah.
So mutations, all that. Like I'm telling you guys, get those questions in. You will get some really good, solid answers right now.
Yeah, I mean, 100%. They're asking if different mutations are brought out under certain environmental conditions. I mean, that's pretty much exactly how they do come about. I mean, extra UV light, chemicals in the environment, stressors, those could all cause mutations. But whether or not those are a germline mutation or a somatic mutation is kind of a different question. If it's a somatic mutation, it's going to only result in certain cells of the plant, and that's not going to affect the offspring whatsoever. But if you happen to have a mutation where the code for the genome ends up being incorrect in the germline, like the sex cells of the plant, those kind of mutations can be passed on to the offspring. But it is definitely brought about by certain environmental conditions. And I mean, that could be artificial, like, adding a certain kind of plant growth regulator, or it could be, kind of naturally occurring where you live at a really obscure place on the planet. Like you live right along the equator, and maybe the UV is a lot more intense there, or you live in a place where it's getting too cold every night. All of those things could bring on mutations. And like I said, there's somatic mutations, and then there's germline mutations. And when you see a plant and there's one leaf that's kind of messed up, that's a somatic mutation. When you see a plant where every single leaf is doing the same exact thing, it's more likely to be a germline mutation where it's being affected throughout the whole entire plant, not just a couple sets of cells.
Okay, let me get. I'm gonna tell you guys on Instagram if you guys have questions, especially like, just not especially anything. Just come over to the YouTube, man. I'm telling you guys, come to the YouTube so I can throw them up on the screen. Like I look down and I notice some questions occasionally on Instagram, but I can't see your guys' questions here. But I'll read yours. So
where are you seeing the questions at, Chad?
Okay, so if you hit the comments, there should be a box on the corner of your screen that says comments. If you hit on that comment thing, it should open up the box of questions. For Instagram, I have to look down at the phone because we could see YouTube, we could see Facebook, and we could see Twitch on here. The Instagram is not like we're on Instagram, but the questions I can't put them up on the screen. It's just the way the system works. But in the corner, you should see comments, banners. I think you can see comments, right?
Yeah.
Hit on that, and it should open up the box.
Okay, cool. Yeah, I'll go through as many as I can, starting from the most recent the most recent ones. Someone's asking if I've done any work with ABC. No, I actually haven't. I mean, I kind of focused most of my energy on the the fern type mutations. I definitely have a pretty good understanding and history of ABC. I know a lot of people that have worked on it. I've seen a lot of the polyhybrids. CSI, Caleb from CSI has worked a lot on ABC. Hive that makes the EXE spray that a lot of breeders use. He's worked on ABC and hybrids of ABC, what she calls sub 1, for 20 plus years. And I'm sure he's still working on it. To me, it was a little slow. The leaves were pretty small, and I have some pictures of comparisons of with the fern leaf mutation right next to kind of the ABC leaf set, and you can see they're a lot different, and you're talking a huge difference in surface area, in terms of leaf size.
Let me throw them up on just read them off the screen, only because if I go down, see the new questions are going to keep coming in. If you're going to, if I throw them up on the screen, we'll get to the end of them like that. Okay. So, did we talk about this already? Triploid tendencies?
Do you have any strains that have triploid tendencies? So you can't actually see a triploid. There's no visual reference for a plant that has 3 sets of chromosomes. You'd have to do what's called flow cytometry. I'm totally butchering the name right now. But anyways, essentially, it's a way where they could look at the root tips and they can see how many sets of chromosomes. They say that MAC1 is supposedly a natural triploid. But there's ways to induce it, and there's people that work on trying to make triploids. It's just not something that we're super interested in. If we ever ran into one that was naturally occurring, we wouldn't really know. I mean, you could assume maybe that there's certain traits that you're looking at, like they say that it has larger buds and certain things, but there's actually no definitive way without testing to know that you're working with the triploid. Germinating.
Let me read the one off Instagram real quick. So, what's the mutation when one leaf grows normal and the opposite leaf grows smaller, disfigured, and completely abnormal growth?
Sounds like a somatic mutation, and a lot of times that happens from older seeds. If you have older seed stock and you're popping it, I don't know if it's from the plant's trying to the embryos inside of the shell, and the older that it gets, maybe some damage occurs to that little embryo, and certain parts of the leaf don't grow out correctly. So you have some cells that are trying to elongate and grow, and some that are maybe stunted in a certain place. So you'll see a deviation in leaves or really crinkly leaves, and those are things that I tend to go away from. I eliminate those all right off the bat if I see that in any of my breeding stock. And it's not from older seeds. If it's from older seeds, then I make the association that maybe the embryo was slightly damaged and it'll come out of it after a few leaf sets and it'll grow out of it. But if you see it right off the bat in brand new good breeding stock, things that you've had just recently done, then you want to eliminate that as much as you can. Just inferior.
He said, what if it never grows out from seed?
If it keeps doing that the whole time, one small leaf and one normal leaf, I would assume still that it's some sort of somatic mutation. I mean, you could send me a picture and I could probably make out what it is pretty quick.
Tips for germinating old beans.
There's a lot of different tricks. One of them being to kind of stratify the seeds. Another, you could scratch up the seed surface a little bit. Some people will like slightly, very carefully clip off the end to try to help it sprout. You could also use tissue culture to do that. You can sprout in, kind of in vitro where, you're in a sterile environment. And then what I've been doing, and it's been pretty successful, there was some germinating solution actually that I tried from a laboratory and it didn't work the way that I wanted. So I experimented with gibberellic acid, and there's a certain concentration. It's about, like a drop per liter, and that'll help your seeds germinate. So if you get some GA3, you could experiment with that, and it definitely helps older seeds germinate.
We have Pop 41C's pre-show, really cool plants. I think that's not really a question. I'll throw it up there though. I had a freak show pop up. Let's go. Have you done? Okay, so we were just there. That's what you answered. Here you go. What are the terps and effects you normally get from freak show?
Freak show is kind of a terpenoline dominant strain, so it's like typically you're going to have that old train wreck kind of smell. The effects are really up, creative, daytime high. I mean, the genetics consist of, big Sir Holy Weed, so there's sativa in there, and like from most of the selections that my dad did on that, it's pretty dominant in that kind of buzz. I mean, you're going to end up with a daytime high, a typical sativa high, happy, up, pretty good buzz actually. Good flavor too, really good flavor. Pretty much, all as long as you have a good pheno, it smokes good, has that kind of terpenoline dominant flavor, down to the end of the road.
I'm gonna say Shadowborn. I think
Shadowborn, fucking the Z Truffles S1s are fire.
and coming soon, right? I think that I think we spoke about this, so I would say within the next week, we're gonna get the Z Truffle S1s. I think there was one other thing we spoke about, so probably we'll just call it next Friday. By next Friday, we'll probably have those on the site for you guys. Bugs can't hide on the back of leaves. Okay. That made sure.
Surprised we're having reception back here today, man. It's so random.
It's great. This is the first time this is perfect.
Normally it's coming out, it's breaking up, but
yeah, totally, bro. Totally. Have you seen plants with triple calyx at the node sites before?
I assume that they're kind of asking the same question the other person did about world phyllotaxy. Unless, throw your comment down if you're asking something different. Bracts growing on leaves, bracts growing right at the kind of division between the branch and the actual main stem. That happens sometimes too. So you'll have little buds that are formed up right at the edge where the actual branch is coming out off of the main stem. That's another kind of mutation. That one's not too common.
Have you ever grown the Dr. Grinspoon mutant?
No, but that was a pretty popular. That was a pretty popular mutation from back in the day that made it to High Times and made its way all around kind of the world where people knew about the Dr. Grinspoon because it had that unique kind of trait where the calyx is just stacked on top of each other, or the bracts stacked on top of each other, where it looks like a string of pearls kind of. And I've seen there was a few breeders that ran into it that was completely, an unrelated strain later on down the road, where they ended up having a few plants that threw nugs that did that same kind of look.
Someone said seems like okay. The whole calyxes would form a claw.
That sounds like foxtailing. I think they might be talking about the Dr. Grinspoon.
Have you ever seen a zymoratic plant? Two meristems out of one bean.
I've definitely seen, two plants come out of one seed. I don't know whether they're asking, one singular plant that has two meristems, two, kind of divisions at the top, or if they're asking, a single bean that has, two embryos come out of it completely. Because I've definitely seen that, and I've seen people almost breed towards it, where there's a whole lot of seeds that are coming out as twins. But also, I mean, it's not necessarily. I guess I'm kind of incorrect in saying breed for it, because it's something where it does happen if you over pollinate your plants. If you just hit them way too hard, two pollen grains will, reach the same ovule, and then you end up. I'm pretty sure that's how it happens. You end up with twins, essentially like a Siamese twin, where they're fused together.
He said the bud itself didn't foxtail. I'll share some pictures. So, okay, wait.
Yeah.
Let me go to, I'm just going to throw that out there. I've seen other plants that could hold 3 seeds in one calyx. This is a second person. So do you want to shoot out or speak out your email? What's your email to send mutant picture mutation?
Email me at cannaresearchco@gmail.com, or you could DM me, on my Instagram.
I'm growing out the Dr. Grinspoon and Cafe Racer. We got the Boro Florist in the house. Let's go. They're up next. So we have about 4 minutes left, 4 or 5 minutes. If you guys have any questions left at all, ask away. Feel free to ask away. Ask away. Otherwise, we can talk about what's coming up. We could talk about the Z Truffles S1, and I think there was whatever else we spoke about. We could talk about that a little bit. Z Truffles S1.
Z Truffles S1, the Watermelon Rolex F2s. I mean, that's something that's just coming out super phenomenal on all ends. The hybrids and the F2s. The selection that I made for the male was absolutely phenomenal. The female side was really freaking good too, and from what I'm seeing, a slight improvement in the F2s over what I saw in the F1s, which is not typical. Usually you see like kind of more variation, but the parents were so stable, the grandparents, and they were so desirable on both sides that I see an array of things that I like. So, but I'm a breeder, so when I look at this, I'm like, wow, I could go this direction. I could go this direction. I'm smelling stuff that smells like, sherbet ice cream with like a Skittle smell in there. And then I'm smelling like a red cherry icy. I smell Starburst. So it's like, those are all desirable kind of profiles, especially when I'm breeding for live rosin and stuff like that. So you'll probably see when I get, I'm already on the F3s, so I have my male selection, I have my female selection, getting ready for the F3s, but I'm going to keep going through there, and there'll be several different versions of F3s, and then there may be some back crosses where I use the male that I used for the F2s to work into the F3s. So there'll be a certain amount of back crossing with the males that I kept. So that's something we could talk about too, is, I've been keeping males alive so that I could work some of these test crosses. And one of the more exciting ones that I have going on right now is the In the Pines. So what I'll do for a test cross is I'll take my plants that I have that are desirable to make the crosses with, and I'll make a smaller pollination, and I'll do 7, 8, or 10 of my top strains with this In the Pines, just so I could get those seeds out and see what the offspring's like. After that, I have the male, and whatever one comes out the best, I'll use that male to make a back cross one, so I can bring the genes back in, and I'll try to work towards a pure In the Pines line, going that direction until I find a female that's desirable, or if I don't, then my only option is to keep working with the back crosses to try to stabilize that particular line that I really want to do. Run out into this. Yeah, the Straupaya Rolex collabs are going outdoors in a couple different depth spots, and they're phenomenal, dude. I mean, the Straupaya, that Fina was like, really candy and really close, and it resembled a lot of the attributes that the Rolex had. So when they got brought together, I mean, I see the papaya side of things come out. There's some that have the pink hairs, the real papaya dominant smells, but the Rolex is there 100% still. So now you have the papaya smell. I don't smell too much that are strawberry, but the papaya predominates a lot of those crosses. I mean, you're going to get a good portion that have the papaya dominant profile, and then the watermelon Rolexes on the back end, which just makes it even sweeter, because some people kind of associate the Rolex smell like with a papaya or something sweet like that. But I mean, if you've ever grown them and you have them side by side, they're a lot different, but really desirable. A matter of fact, when I first found my cut of watermelon Rolex, I said to myself and like a couple other breeders, I was like, this is the only thing. If you were to go, in a competition and papaya is, the top leading thing for like a fruity profile, a candy type profile, the watermelon Rolex is like the only thing that I could think of that would beat it, heads up right there, But,
yeah, and those stuff are on the site. There's still packs left on the site of those, and I think there was only 20 packs. I see Puff say you got 2. There might be, there's some of those packs left on the site, just so you guys are aware of that. Go to the site and take a look.
We'll grab them. Those are so worth it, especially for breeding and hash making.
Hey, Boro Florist, you guys can come in whenever you want. So, whenever you guys are ready, just click on that link. I'll see you come in the back end, because I know whenever you guys are ready, come on in. All right, wait. Instagram just ended. Hold on a second. Let me do this. Let me throw this. Hold on.
Someone's asking. Nah, there's no particular brand. I mean, for me, when I'm getting PGRs or chemicals, I mean, there's some places that I prefer. Plant Cell Tech is a good one to get your PGRs from. But I mean, if you're just gonna get GA3, I mean, you could get it from anywhere. You could get it from like a Chinese source. You could get it from, like a non-branded source, and still it's gonna work perfectly. It's something that's been used for a long time, and it's really cheap, and it works really good at what it does. I think at one point or another there was people that were experimenting with doing reversals or making certain kind of reversal sprays that had gibberellic in it too. Kind of helps the plant stretch, but then again, it's a hormone, and it could cause some certain amount of mutations too. But from what I've seen for helping germinate old seeds, I haven't seen any mutations come out of using it for that purpose. It's a really slow, small concentration. It's not like taking 1 ml and like spraying it on the plant or whatever.
All right, I see you guys back here. Hold on. I'm just trying to get us back on Instagram real quick. It ends every hour, so it takes me a second. Come on. All right, here we go. We should be good there. All right, let me get these guys. All right, so I'm going to bring these guys up. I'm going to have you come back next weekend, maybe, Max?
Yeah, we'll figure it out.
Next week? All right, cool. I appreciate you jumping on, man.
Cool, brother.
I'm going to let these guys in now, and I'll see you. I'll give you a call tomorrow.
Cool, man.
All right, brother. Thank you.
Stay up, guys.
What's up?
Oh shit, how's it going?
She had us muted. Hello.
I was like, wait, I think you're trying to talk, but I can't actually hear you. So
It's
How
my
does
first,
this internet thing
it's
work?
my first time. I'm sorry. Okay.
No, you guys are good. You guys are good. I mean, nice to actually like meet you guys face to face, right?
Yeah, this
It's
is a
so good to
pleasure,
meet you
man.
too. Yes.
Yeah.
Thanks for having us.
Oh yeah, my pleasure for having you guys. Thanks for coming on. I feel like, I was talking about this earlier. We did tissue culture. We had, in-house and then kind of research. And then you guys, it just brings us all together. It really does, like all aspects of it, you know.
Yeah.
So
That's really our kind of our take on it too. Like with us being glass artists, where we've ended up in our part of the community, like we look at all of these aspects on a regular basis. Cass has been working on genetics for going on the better side of 20 plus years now. I've been doing my own research for medicine over the last good long while here myself, having Crohn's and, the need to be a patient. And then just being a glass artist as well could introduce us to really cool places. We get to travel and see a lot of fun stuff. So
isn't that like the best? I love the travel, so it is such a big part of it. Whether it's going to events or going to see a concert, whatever. It's just a huge part of like my life. Love it.
Sure.
But yeah, so where are you guys at now? You guys are up there in Canada, or
We are. We're in Quebec right now, actually.
okay.
Just north of the Vermont border, which is kind of cool. So
Yeah, I have like no clue, like where because I've heard there's so many Canadians at this point, like they're you see, I mess with them all the time, but I love them. They're my brothers, so
I think we're
um,
like 45 minutes from Canadian Bacon Mike.
yeah, I think you guys are. He was saying he thought you guys might be stopping by or something. I'm like, okay, cool.
At some point, yeah, we have
we'll
to.
see which way the wind blows for that one. We might do it. But yeah, I guess we're pretty close, within an hour.
Yeah,
Yeah,
45 minutes.
you guys should go see him. He's a great guy. I'm sure you guys know this already. So
Yeah.
but I didn't. Did we talk about that? You were doing breeding. Like, did I know that? Did we have that conversation at all?
I don't think we ever had that conversation.
No, I don't think we did.
Back in the day. So I actually went to school for anthropology. I have a degree in forensics. And with that came a study and an understanding of genetics. So I have actually coded my own mitochondrial DNA back to my own genetic mitochondrial Eve, which was really cool. And in that, found out that I had Viking lineage, which was pretty freaking awesome. So,
That was pretty
um,
cool.
yeah, it was pretty rad. They came out with a Netflix documentary about one of my actual ancestors, which was pretty awesome. So, yeah, so through that, I actually and like my love of plants and growing and all sorts of things, I kind of moved and gravitated my genetics understanding and education over into cannabis. I also was a propagation manager for an eco-friendly greenhouse and was in charge of 3.5 million plants. It was a floral greenhouse, so very similar to cannabis, but different. And my job was to be in charge of plants from either seed or tissue culture all the way up until they were out the door, which could consist of up to 3 years' time, which was pretty cool.
Wow, how many million plants?
3.5 million plants, yep. All
Plants.
sorts of different florals to pine trees. There were what was really cool is one of my projects was figuring out how to basically winterize eucalyptus and bring it from New Zealand and have it withstand our winters in northern Canada. So it's really cool because there's actually eucalyptus that's in stores right now all across Canada that I actually helped to bring to fruition. And you can very much take it, put it outside in your garden, and it can withstand the whole winter, which is not normal. It is very much a tropical plant. So it took a few years to get that to happen.
Yeah, that's pretty cool. That's very
Yeah.
cool, actually.
Yeah.
So have you done any cannabis breeding at all? Like, are you
I have.
okay?
I am very passionate about landrace strains, and so I have actually been traveling across North America in search of our heritage landrace strains. I have a passion for bringing strains that are like not ancient but ancestral, if that makes sense. So older than essentially any of the seeds that we've come in contact with currently. And I do have this understanding and belief that they are out there and that they have been preserved for us. We just need to find them. And it's really cool because we've been finding, new, not new, but like hidden landrace strains. They're popping up more and more. And it's really like watching my friends and learning. And it's been really interesting to see, how old are these genetics? How can we find if some of them have been, like played with, or can we actually find the ones that haven't been touched at all, that actually are so ancestral that it's almost as if,
They've
you're the
usually
first
tend to
human to touch it. Yeah.
a paleo forensic
Right?
approach
What
to the
I'm
genetics. You're
thinking, yes.
trying to like literally dig up what's been buried
Yeah.
and bring it back. Like Jurassic Park
That's it.
extracting the DNA from the amber.
My prof,
Right?
one of my professors, his dad was actually that human who, in theory, like Jurassic Park is actually based off of him. His dad was the one who found the mosquito in the amber and wanted to extract it. That's why my brain is just like, yeah, we can do this with cannabis too. I don't know. Maybe
No,
I'm
that's
crazy. What do
fucking
you guys
cool.
think?
No, I fucking love it. I love it. I love the idea. What's the oldest seeds that you found so far? Like, how far back have you been able to go? In your search, like how far back?
In my search, I've been very fortunate. So I met the humans who brought strawberry cough to Canada. Can you guys guess how old that was? How long do you think strawberry cough has even been in the United States?
I mean, I would have to say no earlier than like the 90s, I would think. 70, no, 90s, right?
70s. 70s
70?
is right. You are
Okay.
right in the 70s. Yes.
Okay.
Yes.
Okay.
So that was the oldest. The oldest seeds I've come across to date right now were strawberry cough, and they were from 1972. And what was really cool was that my friend had actually started to like pop lineages of them. And so I had generations of them that had been, when I was given them, they were F2s. Unfortunately, my entire seed bank is not with me anymore, which is sad but true.
Your
I
seed
know that,
bank or your
yeah,
seed, like
I
your
had a
personal?
heritage seed bank. Not only
Oh,
seeds, but like I do it with glass too. I like if there's like secret prep or like old, like really old hidden shit that like nobody's seen before, even if it's like a stick of like, I don't know, Amazon bronze, I will like covet that shit or I will search it out.
She has a really weird scrap bin, is what she's getting at. She collects all the weird little dusty things. As someone who likes to study dead things and what happened before does.
Yes.
let's
I
go.
know. I should have called myself a glass anthropologist now or archaeologist in hindsight, but
I mean, I get, I know people that like have definitely been preserving stuff and some unique stuff too. Like, we'll have to, I'll have to see like what I could help you find too, for
that would
sure.
be rad.
Yeah. Like
Honestly, tonic florals, yeah, like it's been in their brain since 2000. I agree. We're looking for ancestral seeds. There are ancestral seed banks. I'm telling you, they're out there. There's people who have them, or like, your great grandmother had a fucking stash somewhere and she was like, she kept it in her cold cellar and you're gonna find it. Like you are. And it's just gonna be like, okay, what did we have? Oh, that's
we love you too. We love you too.
Much love, Mom
Much
Dog.
love.
Yeah, no doubt, man. Like, I am gonna definitely go out of the way to try to, like I said, I have a couple guys that do preservation work, and I
Yeah.
know there's some like, 78 like I can't remember what he has. I think Maui. Wow, he's got a whole bunch of old stuff. One person in particular. So I'm definitely gonna ask around and see what I can find as well for you guys. But yeah,
Well, here's the one that we've spent probably, a solid year, I think, actually looking for is a real lockdown on Charlotte's Web, the pure
Yes.
CBD strain.
You've seen
Now,
me pop in and ask.
the reason we're looking
Yeah.
I'm thinking we might not be able to find it because it got licensed back in 2011 when it was designed. And maybe they are the only people who have it. I've never seen it anywhere else.
I am going to
I've
ask
seen-
around because there's a couple guys even in Michigan. There's a couple of my friends up in Michigan that I will find out. I'm gonna see what I can hunt down for you guys because I just talked to so many people and I feel like we have got to be able to find that. You know, I know you guys probably been looking a long time, but I'm gonna do my best to try to find it for you guys first.
We would appreciate that, yeah, because all the friends that we've talked to in Michigan, around other parts of the states, in Canada even, they've got, you know, lineage of it, but it's crossed in, you know, a way that doesn't really provide the medicine. Like everything that it's been crossed with that Cass has been able to experiment with, it's not what she's looking for to create a medicine with.
I don't want canatonic or catatonic. I don't want that. I
Mhm.
don't want like Moby Dick or ACDC or any of those. And I get that they like all, you know, kind of come from Charlotte. I want Charlotte because I want to create proper RSO for patients, just like Zelda did back in the fucking day, that is actually beneficial for people who have seizures, people who have like restless legs that like goes beyond restless legs for like people like myself with Crohn's, like
Yeah.
and CBDs like Charlotte that are like specifically like I don't know how the fuck it just I know there's a reason why I'm looking specifically for her, and not any of its counterparts. Honestly,
Have
that's
you have
how.
you have you tried the cop? Like, have you grown out and you just know? Have you had the shark? Like, you have you
Back
had
in
that
the
in your?
day, and yes, in
Okay.
Canada, and that's why again I'm upset. Like I had this amazing fucking seed bank, and was very fortunate with my friends, who were, you know, like this older community of folks who had been growing for a very long time. Um, yeah, I ask in the States because I haven't traveled, you know, like we've just been traveling for the last couple of years, and I always wonder. I always ask up here in Canada, and it's been gone for a while. So, it's something that I'm always looking for, honestly.
All right. Well, you got me on the hunt now, and trust me, I talk to a lot
Heck
of
yeah.
people about genetics
Thank you
all the
so much.
time. So if anybody can find it, I have
If
a
you
good
can
feeling.
find it, honestly, yes, I
She
will
wants- yeah.
happily make RSO for the lot for everyone, honestly, pro
That's her
bono,
goal,
like
you know?
that is my goal. I just want to make proper meds for people who cannot access them themselves. It's something that I'm extremely passionate about. I appreciate you guys. Yeah, you'll realize I'm a pretty passionate human.
That's a wonderful thing, man. Like for real. Yeah, I see it. I see the comments. You guys can see the
Okay.
comments. So
I appreciate you guys.
So okay, we talked about genetics a little bit. I mean, listen, we should have started at the beginning and let you guys introduce yourself, right? Because I don't even think we did that. So, you know,
We jumped right in.
you know, the people that are in here that might not know who you guys are, where you're at on you know, where they can find you, like where they can
Heck
find
yeah.
your class, all that. I want everybody to know. So
I'll give you a little bit of a backup about myself. So you can find me on the Boro Florist on Instagram. I've had a million names on Instagram, honestly. I've been on this platform since it started. I was known as the Backwoods Bake Shop. I was also Buckle Up Buttercup for a few years. My first glass name was Evolution Glass, and that's what I was known as in Canada. And then over the last 3 years, I've actually evolved that into the Boro Florist, where now I do all of my work predominantly. I make glass floral bouquets, both functional and non-functional. But my previous life before glass blowing, I've only been blowing glass for the past 4 years. I guess I'm part of the like COVID cohort of glass blowers. I say unfortunately, but it's really not. I used to work for the Ministry of Health in Canada. I also used to work for McMaster University and was a researcher in medicine for multiple years. And during COVID, my job and almost 200 others, our jobs were deemed redundant. And so, I very fortunately enough had some friends who were blowing glass, and they kind of showed me the way. I had never ever thought of being artistic at all. honestly. Like, I cannot draw a stick person to save my life. It's actually the truth. Like,
You make some
I
good
laughed.
flowers,
I
though.
laughed at my friends in their face when they were like, "Here, you should come do this." I was like, okay.
It's
Um,
fun, man. I've
it's
had
so much
you know,
fun.
I've had friends that have been blowing glass a very long time, and I used to go like this is way back when, and they would, I would go over and sit, and they'd be blowing glass, and they would teach me how to do like mushrooms, and I learned
Yeah.
how. But it's like the shaping of pipe, like that shit is not easy, the raking, like these guys, this is a good friend of mine that blew this. Like I've known him forever. He's like my brother, so
Yeah.
I'm very well versed on like ideas of how to do it. But what you do, what he does for sure, like that's like next level to me. Even my
I
buddies,
know.
my buddies that blow like really sick glass,
Yeah.
like they can't really, they're like, nope, you got to find somebody else to do that shit. I'm like, okay, cool. But now I found you. So between the both of you guys, obviously, it's I'm
It's
pretty
true.
stoked because yeah. I mean, I love glass, man. I collect glass. I have pipes everywhere, like always have for fucking such a long time now. That was like the
Yeah.
coolest thing. Like I would go, to shows and we'd see like way back when Bob Snodgrass would have to, Grateful Dead shows and
Yeah.
nobody knew what it was. Like you're like, whoa. And it just became like such an addiction and a hobby. Like I would, it's always been like that for me, like ever since back then. And these guys, they learn with Bob. They were apprentices of his. So, it goes back to him. Like he is the godfather. Like it's cool. It's cool. Like the
I
history.
hope
Would
one day.
follow everything to Bob Snodgrass and the Grateful Dead. If it wasn't for the Grateful Dead doing their world tour and bringing European pipe makers back to the States with them Bob Snodgrass wouldn't have known about it. None of these people would have known about making pipes out of borosilicate.
No doubt.
And
No,
this whole thing would have never kicked off back in the 80s.
It's true.
no, it's
Like-
beautiful, man. Like I'm not, yes, it's just, yeah. And when I see like you got, I see the passion, like I follow you, I see what you do. And I just, and her with the, all that. And I'm just like, it's fucking great, man. And then I mean, people don't realize like when I have guests come on, sometimes the first time we actually have conversations and it's purely like organic. Like it's not like we talk about, we're going to talk about, we like her. Yeah. We'll talk about community, that. But no, like it's just great to like be able to like, first of all, meet you guys and have conversation with you guys, and like such good energy, So
It's an
really
absolute
Appreciate you
pleasure.
having us on.
Yeah, it's an absolute pleasure. For those of you,
Go ahead.
I'm known as Snoopy. I have been since I was like 13, 14 years old at the skate park riding BMX. I've always known about glass. I've always collected pipes and stuff since I was a teenager, being a degenerate, getting high after school and things like that. But when I got Crohn's, or when I found out that I had Crohn's and I was starting to deal with the ailments, and I found the way that cannabis was able to help me medically, was that I really kind of took the dive into what it meant for me, cannabis wise. And then only a few years ago, more recently, did I find glass as like an art form for myself. So I've been more of a blue collar worker. I've turned a wrench in many mechanic shops. I've ran a couple of galleries and things, or ran a couple of maintenance places, done the inside of the dealership, car salesman, all that, and then took a turn and did ironworking. So like as blue collar as you can really, really go, tying rebar together so the concrete workers can come pour the bridge. And that stuff was wild. And, one day my friend says to me, well, I'm laid off for the winter because the weather in Wisconsin isn't the greatest for construction in the wintertime.
Well, I think it's yeah.
Oh,
But buddy's like, come work for me at the smoke shop. You're not gonna make a lot of money, but you'll keep the lights on in your house and you can do basically whatever you want. It's a smoke shop. Like, all right, yeah, whatever. Well, I was there for a year, and then I'd gone to a glass trade show in Las Vegas, and I had never seen anything like that before. There's 1,000 glass blowers all doing amazing work I couldn't even fathom before. I'm like, what? This is a whole world I didn't know about, man. It's like a secret society of this like $1 billion industry nobody talks about. Like, I want in. I saved up some money and bought a torch. I got myself a little space to start working in 2019. Spent a couple hours on the torch between then and there and was like, wow, I really, really want to do this. I quit my job, started blowing glass full time about the same month that COVID lockdown happened.
Right.
So I kind of rope into that whole group of COVID-born glass blowers, but I had the idea before that.
Hey man, we all like found something to do in that point in time. You know,
Yeah,
I think
right.
Yeah.
Well, it's like I either buckle down and I figure this out, or it's back to the blue collar life and see how that goes. I had this passion for glass, and it'd be difficult to try and pursue something like that and have 40+ hour a week job, all this stuff. I had already tried to balance that, so I just kind of went for it. It turns out I kind of have a little bit of a knack for it, I think. People seem to be stoked on my work when I present it to them. I'm stoked on my work. We've done some really, really freaking cool collabs with really cool people in the last 2 years, and just
Even
feel
in the
absolutely
last
blessed.
48 hours.
Yeah, right. The stuff we're working on right now.
I just finished my second bong with my friend in there. 2 bongs and a pipe in not even 24 hours. We've been killing it.
Are you guys at a shop right? Like I remember you saying you might go live from the shop. Is that where you're at right now?
Yeah, we're at my friend Oak Local Boro's studio. He's in the studio right now. So yeah, we'll take you guys in there. There's 100% a seed in here. Yeah, it's creamy. But that's from legal weed. That's
metric bean. Metric bean.
That was a squirrel moment. I'm sorry. I was like, wait a minute, that's a fucking seed.
Little tiny green guy.
That's a seed.
Anytime there's seeds, that's when you're like, man. I don't give a fuck.
For an $85 ounce, I'm not going to complain that much about it.
Still.
No. There you go. Some people that know you will throw that up there.
Oh yeah.
Oh, thank you so much, my friends.
Yeah, man. I think it's I started doing what I did during COVID too. So the seed bank, all that came about in that point in time, and it gave us time to actually figure out what we wanted to do. Some of us, I mean, nobody had to work, nobody had to do anything. So the passion for what we love to do, I feel like it came out. And yeah, it's cool to see you guys, doing that for sure. Do you guys do any collabs together? Like, are there pieces where you guys are incorporating your stuff?
Yeah.
I'm very fortunate to say that. I'm very fortunate to say that all the time. We do take the opportunity and collab. We have a small lineup or a series of glass that we do together, where like you can get a hand pipe made by me with one of her flowers on it or some design by her on it. We have an introduction level of banger hanger dab rigs that come with her flowers on it.
Yeah, we call it the flower pot.
Slightly higher level dab rigs with her flowers on it, and then we've got the really crazy stuff we do. But we make the little things a lot.
That's true.
And we're always working on like new little pendant ideas. Like yesterday, she says, "I want to try and make a wreath. Can you make me a glass donut so we can make it a wreath?" I said, "Yeah, let's do this. This sounds like a lot of fun." And then in the end of it, I felt like it kind of messed it up a little bit. Took a little chunk out, melted like kind of flat. But she's like, "No, it looks cool."
I've decided that I'm gonna weave a glass wreath.
Yeah.
And then
because making a glass donut isn't
much more of a pain in the ass than it was worth. So
hey, that's what prototypes are for.
I'm just gonna braid glass into a wreath with flowers. It'll be fine.
That'll look cool. I think that'll look like I can picture how it'll look. That'll look
I've
cool.
been practicing like how to like it's not easy, but it will look cool.
It's not easy.
So Christmas is coming up. I feel like it's like appropriate, with the wreath.
That's kind of what sparked it, yeah.
Right now we're doing spooky stuff.
Yeah, you should tell them about your spider.
I made a spider out of glass. I don't know what to do with it.
But it's a
His
dead
legs-
spider.
Yeah, it's a dead spider, so his legs are all curled in, right?
Like he's okay.
Yeah.
Hey,
Yeah, you know that was the coolest part of it for me because
It's like half-ass anatomically correct. So like it's an inch long spider. So how thick are the legs really gonna be, right? They're glass. I don't
Yeah.
want them hanging out here where they're gonna get snapped. I was like, let's tuck those in a little bit and see what this looks like.
it's cool though because when a spider does die, that's what happens to it. It looks
That's
like
exactly what happened.
That's how I curled them in.
I feel like Harry Potter. I don't know. I can picture the spider in Harry
exactly like the Avada Kedavra or whatever he says
When
to
you
him.
kill it.
Yeah, the deadly curse.
So, do you guys have a website? Like, how can the people that don't know who you are, and there's a bunch of people on. Well, yeah. How can they find your glass?
Instagram.
Okay.
Right now-
yeah, it's pretty much our primary source. That's kind of like our online portfolio that we're able to work with.
We're too poor to have a website. I'm just gonna be
Websites,
honest.
they just entail a lot. There's so many options if you want it to look okay, present your work well, be able to update it
Yes.
regularly. There's a lot of maintenance fees and stuff like
There
that,
is.
that come in, and so we haven't
Yeah,
pulled the
I
trigger
know.
on that yet. So
I know. I have I know. I
I was
know.
gonna say, I imagine with having a website.
It's expensive.
Yeah, no, like the payment process, all that shit is
Yeah.
the thing. It just is, But okay, so they can look on your page anytime and see all the new stuff that's coming up. I always look. I'll tell you that. I do follow both of you guys, of course.
We have a
Yeah.
YouTube. It's called The Florist and the Pipe Maker, and there are going to be videos popping up. We've literally been taking so much content. So the next few days, there's going to be a whole bunch of things popping on there.
Hopefully in the next few days.
See-
I've been trying to deal with the.
Brady says you should make the spider into a handle for a bowl too.
See, that was originally the idea.
You
I was gonna
should.
put the spider on a bowl,
You should.
but I didn't want somebody to be
It'll
like,
look so
"Why did
cool."
you give me this thing? I'm gonna break the first time I use it."
No.
Like, I didn't want to be that guy. I am doing a slide drop right now,
It's more sturdy than it looks.
so
So on the bowls that you guys do, the hand bowls, like, are you doing like raking? Like how? Like I haven't seen your hand bowls. I've only seen the others. So how's that? Like what?
So right now, you go ahead and explain.
Well, like right now,
you can give them a little sneak peek.
well, I'm doing a slide drop right now. Slides for your 14 and 18 mil bongs. Here's actually one of them that I made, and Cass is like, "Oh, I'm taking this out of the drop." So it's a martini. It looks black right now, but it's actually a red over like a sparkly steel wool color. It's got a really cool milli image, we call these. Milli or Marini, Millifori or Marini.
So who makes the millies for you? Do you make them yourself?
I collect them from different Marini artists. I've made a couple of millis myself. I don't consider myself to be a milli maker, although I can do it. I like to collect a lot of different images, and then in case they can fit people's different vibes, somebody's like, "Yo, make me that Wu-Tang rig or like a Wu-Tang marble." I'll be like, "Dude, I got you." Or like somebody's like, "Hey, can you make me like some Jack Skellington vibes for Halloween?" Like, I got you, man. Like,
So Brady,
They're hard to
100%.
make, though,
We were
right?
at the Bake Turkey.
Like,
They're good homies of ours.
Do you just make like I'm thinking like like the tubes of them, like or
Yeah.
Okay. So
yep.
okay.
So I buy like sections of that. They pull them down to like cane, right? So they call it.
Right.
You'll start with a large image or like a tube, right? And you'll stack glass together to make your image, and then you pull it down.
Right.
Yeah. And I'll buy you. Typically, it's like $5 to $10 a gram is what you can buy milli from milli artists. So you buy chunks of it, and depending on how well you can use it, determines how much you get out of it. But yeah, with the slide drop I'm working on right now, I'm trying to incorporate a lot of different ideas and things. I don't want it to just be like, oh, you make one thing one way and you do it well, good for you. It's like, no, there's horns, there's different angled horns, there's cab marbles for big thick handles on the side of it.
Right.
I'm actually making a water functioning slide. If you can imagine that, there's gonna be water that flows through it. So without giving too much information away.
Yeah, I wouldn't give too much information because I don't think I've ever seen that before, and I can't even I don't know. Yeah, it's hard to even.
He's in the
Take
middle
care
of doing a crazy project right now. That's all I'm gonna say.
All right, all right.
Actually, you
taking one
and
of my
Otis are
recyclers.
collaborating.
Yeah, I'm doing a collaboration with Local Boro here. He gets to dress it up, but I'm making a recycler and I'm making it really small so that we can put a bowl on the top of it
Right.
and an 18 millimeter joint on the bottom of it. So when you go to rip your bowl, it'll be a functioning mini recycler on your lawn.
That's badass. Okay. Oh yeah.
I like to play the mad scientist.
Yeah.
I like to
Definitely
take giant fat rips. Sorry, guys.
She just smoked us out.
What do we got? So, a buddy of mine in Winnipeg owns a glass shop called Bake Turkey. Get in contact with him to carry some of your pieces.
We actually just dropped some pieces with them this last month. We got to hang out with them while we were at Puff Puff Pass in Alberta. And they picked out some pieces and asked us to stop by. Actually, just real quick, show you here.
This is evidence.
Here's the evidence.
Oh shit! Look how that happened.
I was like, y'all know you ain't letting me leave without mood mat.
That's true.
I was
Gotta
gonna
have
say,
mood mat.
I didn't. Damn it! I told you I should have
You
worn
should
that
have worn
shirt
the
today.
big turkey shirt.
I was.
You're right.
Yeah. Wait, did I? I don't think I did.
Yeah, you did.
Oh my god, I did. Hold on.
There's more evidence. Gotta get that swag from the
I
homie.
didn't. I swear, I didn't. We didn't plan this, but you know,
Yo,
shout out.
yeah, I'm just saying, man. I think y'all must have planned this. Like, come on. Yeah.
Actually, they're Canadian. They're actually one of the best Canadian fucking glass shops.
Definitely
Like,
one of the
true
best
talk.
ones that I've seen for sure.
We couldn't find a fucking 10 mil banger when we were in Canada. I'm just gonna say it. We drove from Ontario all the fucking way to Alberta. We could not find one 10 mil banger, let alone a 45 or a 90. Nothing. They gave us. You could find every beat male.
Oh my god, I've seen some of the most heinous things. Like you come into a shop and you're looking for something, and they're like, "This is what we have for you." And you look at the guy and you go, "Homie, the community hasn't touched these items in at least a decade." And he's like, "What do you mean? I just ordered them last week."
Like swings don't exist anymore, bro.
Oh, buddy, I'm so sorry.
Like,
I'm so sorry.
What? Like clearly you do not. And then everything's 14. 14 mil is no, we're looking for 10 mil for dabbing, folks.
I mean, there's a lot of 14 mil dabbing going on.
Yeah.
There is. It is popular. 18s, not so much. So there's no need for those to be in the store.
You saw them though. You saw them. 18 mil cord.
We're talking, a joint the size of your thumb. We're doing your concentrate here.
We don't smoke the same, bro.
No, it's okay. I mean, there's not a good blueprint of like, hey, here's the education on proper consumption for concentrated cannabis.
That's true.
There is no, this is how you do it. This is the easy way. And it's all based off of everybody else's interpretations of what they've been able to experience, right? Like that's how the whole smoking consumption experience goes.
That's why we're trying to.
That goes to like, people going to dispensaries, new, people that are new to smoking, all that. We talk about this all the time because I feel like education is just key. It just
100%.
Yeah.
Now, a question here for you. 'Cause I asked this question to a store owner the other day, and I'll add a little bit of a detail after I get your insight on this. I asked him, I said, well, who do you think is actually to blame for this? Whether we're looking at it legal market or not legal market, who's to blame here for bad products and the lack of people knowing better? Is it the consumer or is it the provider?
Oh, I definitely think it's the provider. I mean, in my opinion, I think it goes both ways because the provider is putting numbers on a label and people are just jumping on those numbers. So no, I mean it just depends which way you look at it because there's so many consumers out there that aren't going to take the time to get educated until they are. They don't know any better. So
right.
I feel like the more that we like as a community can put the word
Educate
out there, educate them, talk about terpenes and not just THC percentages, because there's such a big part of like the consumer market that is going to walk into a dispensary and literally is going to be like, I just want the highest THC percentage, and that's not where it's at. And until they like know any better, which eventually it's on them, but it's not. So it's like, where is that happy medium? I think it's on both of them. I really do.
I would agree with you on that. I would agree with you on that, and I lean a little bit more towards the side of the provider, as you had mentioned as well. Well, it's more on the provider. They should be the one to take the responsibility to try to share that information in the first place. And then it's up to you as well to see if your consumer's into it or not. If they're not into it, that's fine. Poke at them a little bit here and then. Give them those whispers in the back of their mind that maybe they want to learn. They'll come around. They'll learn.
What was the shop owner?
This shop owner told me straight up, it's the consumer's fault. All they care about is a high THC percentage. They don't want to listen. They don't nothing. I'm like, well, have you tried to educate anyone? It's like, well, it's not worth my time. So, okay. I just got a message today from a friend who went into that shop, and he's like, hey, do you have any interest in any of the stuff that's in here? He's closing his doors at the end of the month.
It's not surprising.
That must be the consumer's fault that he's closing shop, huh? Like,
it's like, because they're finding other means out there, and just better, higher quality, kind of like it is, effects, terps, all that. And when they're so focused on THC percentage, and shop owners in particular, like that's what they're buying. That's not. Once people get a little bit educated, and they like go out there and realize that something 13-14% with like the full, the entourage effect, like the full, like a really good terp profile, this is what they're going to come back for. Like they're going to stop looking at the numbers, because and you have all these fucking testing labs that like blow up the numbers a little bit just because they know it's just like that whole fucking vicious cycle. But then once someone gets educated and they smoke something, and they're like, then you become a connoisseur. Like this is where we're at now with, most of the people that come through the lives and this and that. But like some of the average consumers out there, they just don't know until they know. And I feel like, especially on shop owners, I'm not saying it's on like the massive, growers to like do that. But when you own a shop and you have customers that are coming in there, you should definitely like make it a point to not try to sell them, the shit that's the highest THC percentage that's costing you, that you're making the most money off of, basically. You should try to make a point to educate like your customers.
Right. For sure. And I mean, when you're dealing with corporate dispensaries, corporate locations and stuff like that, it's to be expected. And I deal with that on a regular basis. This individual is an independent. So he chose to be licensed and put himself out there against all the corporate chads in his cannabis community. And he wanted to be like, no, I want to pick out the good cannabis and provide it to the community. But he doesn't. Because the way that the system works is the corporate chads have more money, so they get first pick on all the product.
And I'm almost also curious about that. Like, in Michigan, like there's so many dispensaries, and it's like, is there any dispensaries that, you really like? I mean, I feel like you shouldn't shop in dispensaries, period. But is there like any that you're like, okay, at least these guys are doing it the right way? Or is it just like, no, like
few and far between?
I think
few and far between.
I think where you find them is where dispensaries aren't allowed to be. Right? There are these hemp speakeasies, if you will. You can get your cannabis backdoored, but you can buy the THCA in the front door.
Yeah.
But then they get so close to doing a right thing, and they're like, bah, money, CBD.
And I feel like that's what happens. Like, I think that there are some of these guys that do go in there like with all the best intentions, and then it's like, and then the competition. But in some place, like I was talking to somebody about this earlier, and she literally was showing me bags she bought from the dispensary. I'm like, why the fuck are you're in Michigan? Like, why would you even if you are a resident of Michigan, how could you not be able to just, there's so many like the caregivers, all that, but yet you choose, and these people are choosing, I guess, because of the I don't know what it is. It just
how many? Okay, but let me ask you though, how many caregivers are out there in Michigan are readily available and easily accessible, and have patient are not full on their patient roster already, where they can have somebody call them up and have an overage? I think you might find that it's not that easy.
Okay, and I was curious about that. That was sort of the next part of the question, was that because I have seen some of the posts of, however many years ago to now, like how drastically the caregivers have
Yeah.
decreased. And I'm not like well versed on Michigan and how it works there. I've been up there and tried to move there and all that, but it just seemed like there should be better ways and more availability of home growers for people like this person that was on with me earlier, where she's not having to shop at a dispensary.
So
I
I think
agree with you. But I think right now when you see something that is pushing something that are named things like Trap Fest
yeah,
instead,
there's a couple of issues that are underlying this right now.
not bueno.
We've got the legal market really pulling in Michigan. As a Michigan resident, I can attest on this. It is actually difficult to try and get proper meds.
Okay.
And I live with caregivers, right? We grow our own cannabis in there. And being a Crohn's patient, it's actually hard to have a supply anywhere that can keep up with us. Now, we're talking 2 ounces of flour a week, minimum. We're talking 2 grams of hash rosin a day.
If we were to keep ourselves
A day.
educated
That,
proper.
at
for our
$40 a
Crohn's.
gram, which is an okay price from a caregiver, that's $80 a day. That's very expensive very quickly. So to be able to find a caregiver, not only that can sustain it, but that you can work with, that you can afford it yourself too in some aspects,
Right.
it's hard. Yeah, we got things like Trap Fest going on and Big Cloud and this guy and who's what and whatever. And they're paying 20,
Yeah.
30, 40, $50,000 for music lineups and comedians and this, that, and the other.
Yeah. I mean, don't get me started
They
on
have
that
a swap meet.
I'm so like, they literally sent me the pricing to have it take all this, bro, it was like $4,000, $7,000, like, all this crazy shit. Like, I think it's just, they're so about their fucking pocket, these guys that throw the, I don't care what any of them say. I really don't give a shit, How about 99% of them are just about their pockets? That's what they're about.
It's this elitist group of people that think that they can be designer. Like, they think genetic designer
Oh, thank you.
is some crazy thing to be on about. No, you're a fucking geneticist like the rest of the people who've been trying to make medicine for however many years. Just because you bred something a specific way and people liked it does not make you a genetic designer. It does not make you the fucking Louis Vuitton of cannabis. You do not get to charge people $10.5 million for a cut.
I'm so glad you said that because, bro, you nailed it. Like, I fucking cannot stand. I've watched it. Like, if you literally are going to call yourself the Lamborghini and you want to change the whole game and you want to try to charge $2,000 for a pack of seeds, I'm out. Like, I'm out. I will never fucking be that person because, bro, that's just wild. Like, it's the plant. Like, this is what you're trying to do. You're trying to change the whole fucking game, really. Selling clones for $10 million. Like, no, that is not okay, bro.
The most absurd thing ever. You're one love. One love. It costs $10 million, but one love, homie.
Like, you know what I mean? You out of your mind, bro. Like, you fucking definitely twisted, bro. Like, I'm just saying, I tried, I really did up until I was done, and I'm just so done with all that. Like, I just am. I give a shit, bro. I do what I do because I really genetics see, what I do because I fucking love what I do. I sit here on Wednesday nights, Saturday nights. I give people out shit all the time. I give out free seeds, free genetic. Like, I have people come back to me 6 months later. Hey, man, I couldn't have fucking survived. I couldn't have made it. My family couldn't have eaten had you not given me that pack of seeds for free. That's why I do it, man. These other dudes are out there doing it for the fucking wrong reasons. And, it's sad. It's just sad.
Absolutely.
Really. Like, I come from a different time. Like, we all had each other's backs, man. We watch out now, and all this infighting, especially in Michigan, bro. All this crazy infighting and all this shit that goes on. Like, I hear it all, and it's just like I almost, I tried and I really wanted, but there's a big part of me that just doesn't want to even be in the middle of all that shit. I just don't. They all talk shit about each other. It's just, you got one organizer that is not cool if you post something, but the other organizer gets. It's just weird, bro. It's just weird.
Yeah.
Like, we should all be okay with each other.
Or
And
if
it's
you
just,
got something specific, they're all trying to bark up your tree and ask you for something, and you're damned if you say anything for anyone else. And it's like, I'm with you on that. I'm just kind of like, cool, guys, we're going to be on the open road. If we're in the area and there's some friends in a spot, we'll pop up and blow some glass. But other than that, like, I'm not. I've
yeah.
coordinated full events for for groups and organizations that are out in that area, and built an entire glass blowing village and scene and everything, just out of the kindness and passion for doing it. And then to come and find out that other artists that I had brought out there gotten, a $10,000, $20,000 paycheck to show up. And I'm like, okay, I'm not going to be greedy about this, but like, where was my paycheck for setting this thing up? If it's not about the money for me, but if there's money to be had, like, I'll take some time to leave for my work at least. Like,
yeah, there's nothing wrong. Like, that's the thing. It's like, no, fuck that. Like, yeah, that just pisses me off. It just does. That's all.
It's things like,
right.
figuring out the logistics behind getting the oxygen and the propane out there for artists, and having the account with the company, and being able to pay for all the stuff and getting it out there, figuring out how you get equipment like this in the middle of a field.
So maybe just explain what putting on a performance as a glass blower entails for everyone, just so they can kind of understand, instead of like coming at it from the explain to, like,
that's a good idea.
Yeah, in here.
So
yeah,
there's
like, I get
like,
certain things. It's hard to like not just be like, it's just because like I've been it's just been a long time, and I've just seen so much, and so much of it is Michigan. It just is,
Yeah.
As an outsider coming from Ontario to all of these events, I can 100% agree with you. But I think that there's something that we can explain coming from this aspect of it. And that's exactly what I want Snoopy to explain, is what it entails, because glassblowing is very much an art form, and it's a performing art form, especially when we come and do these things live for you.
So like, say a promoter for an event or something hits up some glass blowers, it's hypothetical and using examples in reference here. And you want to show for a 3-day event. You've got music talent coming in. You've got 1,000 to 5,000 guests that are going to be walking across the grounds, checking out cannabis vendors, buying stuff, enjoying the music, having a good time, right? A whole campers weekend. And you want glass blowing for the whole weekend under the tent, torches going, cool collabs happening, people throwing down, having this really cool experience for each other. Nobody takes really into account what it takes and on the behind the scenes to actually make that a possibility.
So
we work in studios, which is a sterile environment. So there's no outside wind, there's no dust blowing around, there's no sand for sure. And to go into an outdoor environment, these are all things you have to deal with, which are going to affect the quality of your work. So to start with, you're thinking, okay, pieces might not turn out the greatest, but now it's the actual logistics of getting there. You need some sort of shelter coverage out there. Okay, so that's like a big tent. That's a rental expense. You need to get steel tables out there, so it's not only a workspace, but it's a safe workspace. We're not talking wooden picnic tables that are going to light on fire if something should happen. A designated zoning area. All these things need to be thought about. Electricity needs to be out there, like 4-phase. That ain't cheap if it's not out there already. Like, there's a lot of things, and then oxygen needs to be provided. You need to have a special account for that.
Not even close.
Several $100, just for fuel expenses, things like this. It all comes together, and somebody has to figure all this out. And the promoter of the event isn't necessarily going to want to be the one to be like, okay, I don't know any of these things, so you take care of it. Well, it totals up pretty quickly for a 3-day event, say 6 glass blowers. Well, your budget needs to be about $4,000 for me to get everything together out there.
No doubt. I mean, yeah, like that's just, I feel like, yeah, it's just a lot. It's just a lot, especially like for you to go do that, and like you said, someone to get paid 10, all that money, and then feel like,
Well,
I
it
don't
makes
know.
sense for someone to not want to pay a lot of money out the gate, right? And be like, well, I don't know if I can pay for that much talent. You know, I'm already paying for this, that, and the other thing for music talent to try and bring people out. You know, glass blowing is a cool thing, but there's no guarantee it's going to bring me an extra 1,000 ticket sales or something like that. So it's like, okay,
I think
what I've done is, you know, we've put ourselves out there to supply everything to do these events before on the grace of like, okay, well, don't charge me the $2,500 it costs to have a booth to vend. Because number one, I'm not selling $10,000-$20,000 worth of cannabis this weekend.
100%, man.
Like, and that's where that booth fee should be charged. And then two, like I have all this extra expense, so $2,500 for a booth plus whatever, you know. Let's find a spot where I can provide an entertainment value for you. And glass art has kind of gotten a little bit of a shadow in at least in this area of like, well, if the person I want won't do it for me, I'll find someone else that'll do it for free because they want the exposure and the clout. That's kind of a sad thing.
Can I ask you, like, at these events, okay? Because I talk to breeders, right?
Mm-hmm.
I'm gonna just call one out, Ross, like Fresh Coast, a friend of mine, right? He went to the whatever event it was. I won't call out the event, but we paid so much for the table. I was supposed to go, I didn't go. He's like, we sat there for 3 days. There were supposed to be all these people. We paid all this money for the table. He's like, I sold 3 packs of seeds. There was more vendors than there were customers. Like, is that something that, you know what I mean, that you guys are seeing too? Like, are you, you know, because I feel like, you know, they want you to buy the tape, like, especially in your guys' situation where you need to have the electricity, you need to pay for the tape, you know, the tent, you need to have all that stuff in order to be successful. Now, you have breeders and other people that could just lay the stuff out on the table, you know, it is what it is. But still, like, when you're going to an event like that and then you're expecting there to be, like, you know, more people than vendors, and then you're finding that's not it, like, that's gotta, you know,
So
it's
it's
gotta,
hard
like,
now. You have to pick and choose. You need to really look and investigate at what other events are happening the same weekend or even month that you're thinking about going to one. And unfortunately, the event holders aren't thinking about things like that now. Everybody's looking for money, and they're not actually looking about it from even the patron's point of view. If you're charging everybody to come in the doors for all of these events, how am I supposed to do it? How am I supposed to even do 1 or 2 of these events, let alone every single one of them?
Mm-hmm.
And then you split up all of your vendors as well, who typically, you know,
2 years ago, it wasn't an issue. There was an event every weekend in Michigan, but it was a different event and they weren't overlapping.
right.
I think enough
did they charge
people.
for people to get in the door?
I mean, yeah, there's always been a small ticket fee, you know, a small fee to get in the date or to get in the door there, to get in the gate. And, you know, it's the cover charge to come in. But it's never been drastic until recently. And I think just enough people have not seen eye to eye on where the money's going in these events that they've all separated and started their own things to get that money.
Right.
And they've all decided they know what the hot weekends are for each other. So now we've got 4 or 5 events going on the same damn weekend in different spots of the state or even different states. We've got Michigan vendors setting up Chicago events.
I've been seeing that.
Like,
Ohio. I've been seeing
and
it. I see it. I see
yeah,
what goes on,
man.
bro.
And
Like, I see it. You know, I'm friends with all
so
those guys, so it's like I
there's
do hear
only
it.
like 500 diehards in the Penn State area that try to go to every event. Okay, so if you've got 5 events going on in the same weekend, you maybe have 100 patrons if you're lucky. If you
Right.
split them up good. If somebody's got a better lineup than you have no patrons.
Yeah, and that's just why it just, you know, yeah, it's wild. It just is. And it's sad. Like, you know, it's sad to hear that you have to pay a certain amount to walk in, and, you know, people don't want to spend, you know, you walk in, a lot of people are coming for one thing or another, and you have to pay to get in and buy tickets and just like I said, you know, if you look back, it was about 3 weeks ago. I had T-Dog in here with Forefathers class.
Yeah,
We had
good
the
friends of ours.
you know, he's the homie. He did my logo. This is back of me that you see. He did that, and you know, him and Forefathers class, and we had the same conversation. And we have, we all agreed on it. Like, no matter who you're cool with and your little cliques, it doesn't matter. Like, this is just what it is. Some things just need to be done differently. They just do. That's one of them. But they can't get along. They butt heads. All these, especially the Big Cloud and other. These guys just whatever's going on.
There's one event that I would say their intentions were right when they got started. There was a cover charge to get in, but it was low. And if you had a medical card, it was free to get in. If you're a medical patient, there is no cover charge. That I agreed with. All the stuff that's going on out the back door and behind the curtains of that event, though, I don't agree with. That's the stuff that's going to bring down the cannabis community. That's the stuff that makes cannabis. It's what's been making it a part of the war on drugs still. Like, come on, man. Chill out.
I came up the Hash Bash thinking like, you hear about it. It's been around so long. And like the outside was wonderful. Like, but once we got into the hotel, like that after party thing, I was just blown away by all the garbage. Like, you're like, oh yeah, we're going to see all, bro, like 99% of it was straight like trash and like these dudes out of Detroit with bags of weed trying to sell this. Like, I was like blown away by what I saw there. I don't know, man. Like, I don't know.
Snoopy's gonna go walk over to Oak Local Boro and show you the studio and introduce you to Oak.
All right, cool. But yeah, it's just, yeah.
Thank you again for having us here. We really appreciate this. It's been really fun.
Definitely. I mean, I'm definitely gonna have you guys back too,
Thank you.
So, all right, I see him coming in. I think he's trying to connect. It says we'll need to connect their account. Okay.
I set it up on his phone, so he should be able to get it to go.
All right. So, if you guys have any questions at all, I see a lot of people chilling. Like, there's 60 people in here. There's a few people on Instagram. I tell you guys on Instagram, come over here, because I can't see your questions. But if you guys have any questions at all for them, feel free to ask now, because we have been here an hour already, and I know I only asked them to be here an hour. So, but if you guys have any questions, please feel free to ask. Harry series. Let me see. Last time was 2018. Yeah, like I said, the outside was cool. I was just a little bit disappointed by, the inside. That's all. But a friend of mine threw a breeder's ball. That was fun. That was a good event. It was small. It wasn't, that was the best event I've gone to in Michigan, I think.
I've never been to that one, honestly.
Yeah, I think I talked to him. I think he's going to try to put it together. It's a little last minute now because it was I don't think he's going to get it together this year. So hopefully next year he does it again.
Okay. I'm hoping, well, we'll see. We were planning to be back for September. Have you ever been to the Nest Creek? I've never been to that one, no. We were going to go to Booney Bash, and then we never made it this year.
So, I mean, how are the events in Canada? Like, is there events that you've gone to up there? Like, how it's a comparison to the Michigan events? Because, like, when I go to New York events, the vibe is just great. Like, I think it's just a little bit different. Like, I've had way better times at New York events than I had Michigan. I don't know what it is.
The Canadian events are fantastic, honestly. Ontario, very similar to Michigan. The size of the events, very similar to Michigan. We went to an event in Alberta this year, and it was a smaller event compared to like the size of events. Typically that would be, in Michigan Snoopy's from Wisconsin. We've been to a few events out there actually that have been pretty sizable to anything that we found in the West. So we're looking forward to going to Ohio. I haven't been to one there yet.
Yeah, here I hear, they're new. They're new.
Yeah.
So
we're going to Toledo in November, so that'll be cool.
I see him trying. I think I don't know if it's letting him connect because I can see it. Could you see it? I don't know what you guys see on your side. Can you see at the
Oh,
bottom?
our side's really weird, honestly. I can't see shit on this side.
Oh, okay. Yeah, I have no idea what y'all can see.
Like nothing. You have to click in separate. Like I was just kind of messing around with it in the bottom, and it like separates everything. But if you want to click in and see the chat that's going, it completely locks you out of like the whole main screen. That's why I
Oh,
was
it does?
wondering, like, if I'm down here, can you actually see that I'm messing around with this or no?
No, you know what you can do? I think if you close, okay, so I think you might have it on full screen. I think if you click on where we are and make it smaller screen, even though we'll be, we're smaller, you might be able to
Yeah,
see
I
everything.
have it like that, but it's just
Oh,
not showing me any. Yeah, I have to mess around with this thing
okay.
to get it too. Yeah, this is my first. I should have. Yeah, next
Yeah,
time.
yeah. Me, like I said, I can't see it from the other side is the problem. I don't know what it looks like from your side. Here, wait. I got to
Have
restart
you
the
ever
Instagram.
used like Zoom or anything like that before?
No, I have not.
Okay,
Nope.
because it looks very similar to Zoom.
Okay.
Normally, though, it'll load like your chat so you can like see it live. But this one in StreamYard, you actually have to like go into it. At least on my phone, which is it's kind of weird. What's going on, everyone? I'm sorry. I love all of you guys. I can't read any of these damn comments.
I'm trying to get us back on Instagram. I only have hour lives because I lost 3 pages in
That's crazy. I watched you the one night. You lost 2 in the one night, or was it 3?
Yeah, I lost all 3 of them within like 24 hours basically, and that's exactly what happened. And now I just got this page back.
Right.
Last night, like 3 o'clock in the morning, the dude's like, "I got your guys' pages back," and I was stoked. But of course, my lives are only 1 hour now, so now I have to restart this every hour. And you guys don't realize it, but what I'm doing, I can't see you guys right now. I'm trying to get us back.
Yeah, because you're trying to. So it's the same when you go in and read any of the chats or anything and hear any of the comments. Yeah.
It's annoying. That is annoying. All right, here. I think we're back. Let's get us back on Instagram. It just takes a second. All right, there we go. There we go. All right, now I can see.
Now, can you see him in the background yet?
No, it says device. What I see is devices not connected. They will need to connect their mic cam before you could add them to the stream. I don't know why. He should just be able to go right into Instagram and click on it.
Yeah, I set it up that way, but maybe it disconnected.
Oh, wait, here he is.
There you go.
I
Bam.
spoke to him.
I figured out a thing.
Let's go.
It was playing with the permissions. It opened it up through the Instagram link, not in just a browser. I had to reopen it in a browser and give it permissions to the camera and the microphone and everything. But we're here now.
There you go.
We are here. We are here.
We are here.
Live. Yeah.
We are live, and this is what's called the Canadian Starship. If you're familiar with the glass scene, there is another starship in the States, which is a huge glass blowing facility where absolute magicness happens. So our friend here, Boro, has been a huge part of that and brought some of that love and vibes into creating his own space out here in Canada. So the Canadian Starship is a place where insane magic happens way up north. We'll do a little introduction here. This is our friend Oak here. How you doing, guys?
11, motherfucker. Right back at you, fool. Right back at you.
So perfect.
Portal opens. So this is Dark Start Genetics here. He is a geneticist and runs a really excellent seed bank. He's about getting proper medication to people that need it, cannabis to those that are looking to grow it, and sharing information with everyone in this community. From what I understand, I think it's a good way to introduce you, my friend, to
Amazing. Pleased to meet
I
you.
appreciate that, man. Nice to meet you as well.
Yeah. And then Oak here, that's, as you heard a little bit from us, he's an amazing glassblower who's been in the community, I want to say, 20 years? 15
for glass blowing.
15 years for glass blowing. Definitely got his own take on things. Doesn't let anybody tell him what you can and can't do with glass. And it's an honor to be able to sit here with Cass, to be to have her introduce us to him, and to be able to work here together with them. This is some of the stuff that we do here. Like, how do you turn this camera around? How does that work?
Hopefully you can turn it around.
Is that a thing? Here, let's do.
I don't know if it's a thing.
I mean, I would think you're on a mobile device, right? I would think you should. Oh, I don't. You know what? I have seen people do it that have come in here, so I think it's a possibility, but I'm not sure. Oh, look at that! Come wait here. I can put you on full screen. Watch. Her and I will be down here for a second.
That's
Oh yeah, that's
This is a
wicked.
piece that all
Oh,
three
yeah.
of us made over the winter. So this is one of my recyclers, styled with Oak's fish and a bunch of coral attachments by Oak and Cass as well. The Cass did like all of the coral and like the seaweed, and then we've got a couple of jellyfish implosions and some really cool swirl cane and things that Oak threw on there as well to kind of fill in some of the void space that we've got. In fact, move her a little bit here. Bear with me, guys. So much movement. Give you a little bit of a function view. So this is a recycler. The water is in the main chamber down here in the bottom. When you hit it, it bubbles up and goes through these tubes here and then makes a vortexing funnel in the cone section and then continues to drain back into the lower chamber. So it's a full cycling of water there.
Sick.
We've got, I don't know, like 4 days, 3 people straight working at it in here.
I love the colors on it too.
Wait till you see the next one.
Yeah.
Do you have that? Do you have the flower piece one there? The blue one?
Yep. So we've got the 14 mil flower piece here. This one is all aqua and sandblast. It's got a bunch of Oak's teeth. Comes with a 4-hole 14 mil bowl. Will also work great as a dab rig. It's got his signature. He calls these Fukushima fish.
Let's go. That's pretty cool.
Just
Hell yeah.
super rad, like really gnarly big tooth jawbone on them, and the eyes popping out. Really love these. And then one of the more recent pieces to come out here today. This was a creation yesterday with the Boro Florist and Oak.
Oh yeah.
This is their first bong collab that they've done together.
So she did. So you know, what did you do on there? Oh, they can't see. They can't see you. I can only see.
Oh,
you,
myself? Oh, all of the floral work. So all of those attachments that you're seeing on the bottom, all of the leaves and all of the flowers, those were all done by myself.
Oh yeah.
And the bong itself is by Oak.
Yeah, well, that's sick. I mean, I still need to get with you over what we talked about earlier. Like, I know I missed you and stuff, but I do. I still, yeah, we're gonna get that. We're gonna get that done. Just know that I haven't forgotten.
Heck yeah.
Oh yeah.
This is a super awesome piece. It was crazy to watch these guys make this.
Yeah, that was yesterday we made that one. The day before we made a flower pipe.
Yep, I got that guy right here. What's really fun about this piece is it's a bunch of flowers on a pipe for a flower pipe. But this is also
who would have thunk it, right?
right. This is also actually Oak's first colored piece that he made 15 years ago.
Yeah.
No
Pulled
shit.
this out of the vault and said, "Hey, Cass, let's collaborate on this." So that was pretty cool.
That's what's.
I made all of those flowers, 2 days ago.
They're beautiful.
I try to make each flower unique to itself. I don't really ever create or recreate flowers, honestly.
I was going to, and she was like, "Well, where's that flower pipe?" Of course.
Sorry, I'm sorry.
So here's what Oak is a little more known for. These are his Fukushima fish. I was pointing out on the one rig. This is where we got the idea to start kind of finding new ways to incorporate these with my style as collaborating. You've got right here, the front fin comes out. That's your joint with Dewar seal for a 45-degree 10 millimeter banger. Mouthpiece up here in the tail.
Oh, yeah.
then when you put
They
it
go
away, the little spike goes
ridiculously
back
hard.
in there to finish the design for it.
Yeah, that's pretty sick, bro.
Then for my art friends that are familiar with like the deep art community and the cannabis culture, the skate community, and going like psychedelics, yeah, so many communities.
Psychedelics. Let's
There's a
go.
name some of us have heard, Chris Dyer,
Yeah.
and he has done some incredible artwork. Well, he's very good friends with Oak, and Oak recreated one of his murals in glass. And this is it.
Damn. Yeah. The tongue.
Galactic dude.
This is the Galactic Dude
Damn.
by Chris Dyer, reimagined in glass by Oak Local Boro. And it's fucking sick.
That is sick, bro. Like, look, that's sick.
I can't even begin to tell you. Okay, it looks like an okay sized piece, but the amount of work that's here, the amount of thickness on all of these attached, this is about 3 pounds of glass in my hand. It is so heavy. This is such a heavy, solid chunk of the rig. And it's a 10 mil 90 degree. It's ready for getting the proper hashtags about it.
Yeah. Oh shit! Can they? I don't. You know, you don't have to talk pricing, but if someone could be pointed to where they could look, if you want to talk pricing, that's all up to you. You don't have to. But
Does Oak have a website?
Do you have a website? I know you've got a gallery. He's got a Instagram and an Instagram gallery.
Website will be coming soon.
But yeah, you can hit him up directly for his work at Oak Local Boro, as we've got listed here, or you can hit him up directly at Local Boro Gallery on Instagram, which is just a gallery of all of his works and his collections of glass
And people that he's got the shop also.
You know, others that come through. So, website coming soon. Typical glass blower saying.
Yeah.
The website's coming soon.
Yeah, website's coming soon. I already know. I told you, man. My shit, yeah. It takes a long time. It's a lot of work. It just is. I mean, once you get it set up, and you could just, you know, like I use the same listings. I'll just copy it. Once you get it all set up, you're good to go. It's just a matter of, you know, putting the time in to get it done. But
So this is kind of the whole studio setup here. As you can see up at the top section, this is our hood. This is to capture all of our fumes and things and to be able to pull it out through the ventilation so we're not breathing anything toxic in. And then this beautiful giant bench that we call the Starship. Oak's workstation with his masterful torch, and then all of his guest locations out here on the bench as well. We've got the Boro Florist working over there right now. I'm currently working here, and then he's got some rental space for an OG over there. We've all just, this is where we get out and we vibe and make some awesome artwork together, guys. Welcome.
Oh yeah. Yeah.
Oh yeah.
I think she must be walking in there. We just lost her.
Oh, we lost her. Oh
Yeah.
no. Oh wait, I think I seen her.
All right.
I was saying she probably just walking in there, I'm sure.
So, we forgot the show.
Where is she at?
Damn. Yeah, I'm just really stoked to be able to have you guys come on, man.
It's
Like,
been an
really
absolute pleasure, man. I mean,
Yeah.
to be able to kind of find a spot where the community does sit down and kind of share ideas and passions about things has been it's been really cool. There she
It
is.
just dropped me. I'm sorry. I was like, what happened? It kicked me out.
I thought you were going to walk over there for something. Like, oh, she probably just going over there. So
No, I was just no. Sorry.
Yeah, it's been really cool to just even thinking about, you know, coming on and chatting with you this week, you know, what we could chat about. It's been really interesting because, you know, Cass and I, we have so much passion about this community that we're all involved in, and, you know, not necessarily just being involved in it, but trying to do right by it, not necessarily by anyone specifically, but by the plan, by the community.
yeah, like that's what we do. That's what I do. You know, I think it's a huge thing, man. I just do. Like some of the conversations we had already. It's just nice to meet like-minded, people. It's not often, like, it's just not like I come across so many people, and I mean, a lot of the people that come around my lives, like, I've been doing this a long time now, so we have a pretty good community. But outside of like the lives, I do see a lot of people. It's just, it's just really nice that it's really nice to have you guys on. It's just been a really a pleasure tonight. It just has,
It's so much fun.
Yeah.
Sorry that we went over your time, but we appreciate you so much.
Oh man, I was glad when you said you guys were coming on last, because I figured once I knew he was coming too, and it was going to be both of you, I figured, we'll go as long as we need to go. But yeah, but I feel like I definitely want to have you guys come back. There's no doubt about it. There's just so many things we could talk. I feel like I could sit here for another 45 minutes, another 2 hours, and just sit here and talk.
I think it'd be great if you'd love to have us. Maybe we could chit chat a little bit in between about a couple of topics we want to be, hyper focused on, go into some depth with the time that we've got on here, and maybe a couple of times we come say hi, talk about a couple of things, try and fill in some of the knowledge gaps that we like to help share.
Yeah, like I would love to have you guys come on and maybe be on the, just maybe blow some. Like I feel like there's just a lot we can do. There's a lot that we can do right here with all the people, and yeah, I would love to do it with you guys.
That'd be awesome.
Likewise.
All right, well, let's do it. So, I guess we could say good night. I guess we could say good night to everybody, man.
All right.
So
thanks for sticking with us, guys. Thanks for letting us have Oak come in.
Thank you, guys.
Thank you, Oak, for letting us
Nice to meet you.
be in your studio.
hell yeah. Yeah. I'm just gonna say, love you guys, man. Love you guys, like for real. Such a good energy, man.
It's so much love, brother. So much love.
Absolutely, bro. So both you guys, man, you both coming back. That's all. You both coming back. I appreciate y'all. You guys have the best night, and you hit me up tomorrow, and we'll talk. We'll figure out some things for the future. For sure.
Absolutely. We'll get to chatting.
Bye, everyone.
Much love, everybody. Take care.
Much love. Bye.
All right,
See
guys.
ya.
Everybody else, y'all have a good night, man. I appreciate everybody for hanging out tonight. I know we went, a little bit late. Such a good night. I hope everybody had a really good night, as well of a night as I did. And I'm just gonna say, much love. Y'all stay safe, and I think that's pretty much it, man. So thank you guys. See you next Wednesday night. See you guys Saturday night. And I might do something with in-house after this. We'll see. But good night. Later.